All right. Good morning, everyone. My name is Nonhlanhla Kalebaila from the Water Research Commission. Welcome, everyone, to this two-day event, which is part of raising awareness on antimicrobial resistance as part of the WHO World Antimicrobial Awareness Week. So we have this event over 2 days, today and tomorrow. So hope everyone is going to stay with us. So we have also distributed the program that we're going to be following over the 2-day period. And up in our agenda, I am just going to introduce Mr. Jay Bagwan, who is going to just do the welcome from the Water Research Commission. Over to you, Mr. Bagwan.
Good morning to all the participants and the dignitaries on this program. It's a privilege from the Water Research Commission to welcome you all to this very important event. You know, AMR should not be taken lightly. And we've seen from the recent pandemic that if we don't give attention to this whole range of emerging pollutants, the consequences will not be very dissimilar. So I think it's important what we are doing today and the effort we are putting in from a research side in terms of understanding the AMR problem and the consequences it's going to have, and then also bring in all the mitigation and treatment options that it doesn't get to that stage where we are firefighting. Now, you know, COVID-19 has taught us a lot about it, but it also, you know, created many opportunities. You know, the wastewater-based surveillance that we have accelerated and used has organized this wonderful platform to bring water quality and you know, the monitoring of AMR through the wastewater streams back into the spotlight. You know, it organizes for the future to use the platform that we have created around, you know, surveillance as the monitoring and management tool of the future. Now, we sit in this discourse of climate change where the quantity of water gets more, you know, airtime, but people are forgetting that the quality is equally important. Okay, and if we mess the quality up, the quantity would not be of any relevance. And it takes us back to our own trajectory as the Water Research Commission, that when we were established, our focal points were balancing the two elements of water sustainability, the quantity and the quality. And I think, you know, it's important we start driving the area of water quality management significantly in this very water use environment that we have created, and build the next cohort of skills, professionals, and you know, technologists and support people to ensure that we can manage water quality going into the future. So it's a very important event, and you know, I'm really glad that we are having this event and giving the kind of impetus and acceleration to one aspect of it and a growing problematic aspect on AMR. So wishing you 2 days of fruitful discussion. And, you know, I would also like to thank the Noordwes-Universiteit and other partners in supporting this event and hosting this event. And hopefully we will get some direction, you know, at the end of it in terms of growing our own research and innovation footprint around AMR. I now hand you over to Professor Jeffrey Mphalele, you know, to take us through also welcoming from the university. Thank you very much.
Much, Jay, for those warm welcome. I am going to hand over to Dr. Molale Tom to introduce Professor Mphalele. Over to you, Lesego.
Good morning, ladies and gentlemen. I'm Lesego Molale Tom, and I'll be introducing Professor Jeffrey Mphalele. He holds a doctoral degree in medical virology, a master's degree in virology, and a BS honors degree and a degree in microbiology. His resume is a shining testimony to his excellent work in the field of research and his contributions specifically in the scientific medical field. This has led him to serving on the Ministerial Advisory Committee on COVID-19 vaccines in the Ministry of Health since 2020. Prof. Jeffrey has also published in various research journals on crucial topics such as strengthening immunization services and policies, as well as the vaccination control of infectious diseases. He's an active and expert member in the scientific committee and serves as Vice President for Research at the South African Medical Research Council since October 2014. He has also been elected as a member of the Academy of Science of South Africa and has been acknowledged as a C1 NRF rated researcher since 2017. Ladies and gentlemen, I hand over to Professor Jeffrey Mphahlele, Northwest Chancellor for Research and Innovation.
Dr. Lesego and the Master of Ceremony, thank you very much for your kind words of introduction. I hope you can hear me, colleagues, because you never know with the media, with the technology, if you are audible enough. Can you hear me?
Yes, we can, Prof.
Okay, that's good. All right. I will try to be short because we certainly don't have luxury of time. But just to say that it is always a pleasure to be in the company of scientists. Not only you look forward to learn and expand your knowledge, it's also an exciting opportunity to interact with the world's best minds. So thank you very much for inviting me. Thank you to the organizing team under the leadership of Prof. Carlos Bezuidenhout for inviting me to this auspicious occasion. So it is my honor and privilege to welcome all esteemed guests and participants to this visual symposium. We are here today, colleagues, because the world is commemorating Antimicrobial Awareness Week. As we all know, is commemorated every November. And this is because resistant microorganisms have become the most significant public health threats globally. While antimicrobial resistance, or AMR in short, became so famous due to antibiotic resistance bacteria, AMR is now seen in all microorganisms. So you can name all microorganisms, fungi, parasites, and viruses. And often it happens because microorganisms harbor resistant genes from either the environment or in clinical settings in response to application of antimicrobial reagents. So it's just one way for microorganisms to ensure their survival. So it's not surprising that in May 2015, the World Health Assembly endorsed a plan on antimicrobial resistance. To date, this action plan has 5 strategic objectives. Just to recap, the first is to improve the awareness and understanding of AMR. The second is to strengthen surveillance and research of AMR. The third is to reduce the incidence of infection. The fourth is to optimize the use of antimicrobial medicines. And the last one is to ensure sustainable investment in countering AMR. So clearly these 5 strategic objectives are very, very important to adhere to and to make sure that we do research to support them. Since 2015, the World Antimicrobial Awareness Week has been a yearly global campaign, and it aims at increasing awareness on AMR. And we know that this is critically important because since the discovery of the first antimicrobial, to be specific, antibiotic, penicillin, by Alexander Fleming in the 1920s, there's been subsequent development of antibiotics, and unfortunately, these antibiotics are now at risk of effectiveness because of the misuse and abuse of antimicrobials in general. You will notice that, in fact, if you look at the behavior of us as the general public, we tend to give those who are really the custodians of antimicrobials a lot of pressure, demanding, the antibiotics, even if it's not necessary to be treated with antibiotics. So obviously, the doctors end up prescribing these antimicrobials, but in fact, if we can try and look after ourselves when we are sick, without necessarily, demanding antimicrobials, we can actually limit, the use of antimicrobials in many ways. And by doing that, we will preserve their effectiveness, and we will ensure that they are not put at risk. But as we speak, the world is at risk of antimicrobials, and we know that the antimicrobials are considered amongst the most important advances of modern science and medicine, particularly when considering the millions of lives they have already saved. But as I said, the abuse of antimicrobials have led to the evolution of drug-resistant pathogens, and as a result, antimicrobials are fast approaching the end of their effectiveness, which presents significant risks to human health. So the World Health Organization has declared AMR one of the top 10 global public health threats, which requires a global response. And this is because AMR exists in humans, animals, food, and environment, as the other speaker has already indicated. So we live in a world which is actually advocating for One Health approach, and we hope that as we increase awareness on antimicrobials, we will do this in the framework of One Health. So we know that the World Antimicrobial Awareness Week, the theme is about spread awareness and stop resistance, and this calls on collective action of One Health stakeholders, policymakers, healthcare providers, civil society organization, and the general public to be EMR awareness champions and advocates. So I just hope that we will do just that. As I indicated, that quite often it is us, the general public, and the patients who are the real culprits when we go to our healthcare providers demanding the antimicrobials. So in closing, colleagues, I just want to say that the Northwest University Microbiology subject group, both on the Posh and Mafikeng campuses, is at the forefront of antimicrobial resistant research, combating and creating awareness on the threat posed by EMR. The EMR research is largely funded by the Water Research Commission, and the Northwest University as an institution is so grateful for this financial support. For the past years, the microbiology subject group has hosted successful public and academic lectures, as well as local community engagements on antibiotic and antimicrobial awareness. This year, this message was amplified to the public by means of live radio interviews, SABC News inserts, and research. So we are also fortunate that this year's event is co-hosted by the Northwest University Microbiology team in collaboration with the Water Research Commission, University of Johannesburg, and Stellenbosch University. So thank you very much, colleagues, for being co-host of this event. And I'd just like to thank all the presenters, participants, and event organizers. And I wish you the best meeting and look forward to an insightful two-day program. I really wish you the best deliberations for this meeting. Thank you very much. Over.
Thank you, Prof. Jeffrey, for that wonderful welcome address. Next up, ladies and gentlemen, we will have our first keynote address that will be presented by Professor Carlos Bezuidenhout. Prof. Carlos Bezuidenhout is employed at the Northwest University as a professor in microbiology and is currently the director for the Research Unit for Environmental Sciences and Management. His post-PhD career has focused on various aspects of aquatic microbiology, including surface and groundwater quality, as well as aspects of water treatment. He has over 22 years of experience, has supervised a large number of postgraduate students that are active in the water sector in South Africa and elsewhere. He has also published numerous peer-reviewed publications in his field. Prof. Carlos and his research team completed several technical reports for the Water Research Commission of South Africa, including work on AMR in surface and groundwater, particularly also in drinking water. Welcome, Professor Carlos.
Thank you, Dr. Lesego, for that kind introduction. To the dignitaries and to all the participants, thank you very much, and thank you for this opportunity to present this first of the keynote addresses. Prof. Jeffrey and Mr. Bhagwan, thank you very much for the kind introductions and for setting the scene in terms of our deliberations over the next few days, or 2 days. So I've put the title down as the World Antibiotic Awareness Week 21. So what is in it? And I'm asking that question, what is in it? Why are we doing it? So from the speech of Professor Mpahlele, he already highlighted, as well as Mr. Bhagwan, that it's a World Health Organization initiative, and this year's theme is to spread awareness and stock resilience. And the calls on One Health stakeholders, policymakers, and I specifically put it out, policymakers, healthcare providers, general public, to be antimicrobial resistance awareness champions, and that includes all of us. So the message that came through is that the predictions are indicating that AMR in Africa is a looming disaster. Currently, worldwide, deaths annually is in the order of about 700,000 within the entire global population, and that by 2050 it will escalate to about 10 million individuals annually globally. So what does that mean for Africa? With the predictions, it indicates that more than 4 million people will be dying in Africa by the year 2050 if nothing is done. So that's a huge portion of deaths if we don't do anything. The other aspect is the economic aspect, and that is the current cost of healthcare. And we all know how expensive healthcare is, whether it is in the public sector or whether it's in the private sector. It's huge, and that will escalate enormously if there are no interventions with insofar as AMR treatment is concerned. So it will become, medical health will become unaffordable for most Africans. But there are various initiatives, and I will focus a bit on some of the initiatives that is taking place. If we consider, and that is specifically where the Water Research Commission is working, is within the water sector, because that's a global linker. It links industry to communities, to food production, to energy production. So that is that global linker, which, I mean, we cannot get away from. And so if antimicrobial resistance and these drivers occur within the environment, specifically within the water, then it is passed on and eventually will affect humans. So that is why this One Health approach is so critical, that we do not see the various aspects of AMR in isolation. We've realized it, and there's work that is being done currently to ensure that we see the AMR within this One Health, and that if it's in the one compartment, then it will definitely affect the other compartments as well. So human health is definitely related to what the animals and the plant health, as well as the environmental health. So from all the reports that we can gather, there are some main messages that are coming through, and Mr. Babon has also referred to the quality and quantity of water. So we need good quality and good quality water in sufficient quantities. That's essential for health, for food security, for water security, and also for energy production. But organic and microbial pollution of rivers and dams in Africa is a major problem. And there's a lot of research that is happening, and it is describing how the pollution is occurring and the impacts of those pollution. And wastewater is, loading from wastewater is where some of the main problems lies, because it, from wastewater, pathogen loads within our water system will occur, but there's also these chemicals. And just now I will get to some risk assessment approaches that I use, and it's sort of focusing on pathogens and focusing on chemicals. But something that's been neglected for a long time is the antimicrobial loads, specifically coming from wastewater treatment plants, but also from agricultural return flows and from within communities, because of our the way in which we dispose of antimicrobial substances into our systems, sometimes into natural little rivers or just urban runoff, and all of that are contributing towards these antimicrobial loads. So consumption of the water is normally what is the concern, direct consumption of water, and most of our efforts have been on describing the risk associated with consumption of polluted water sources. However, something that we tend to forget is also that during recreation, there's an opportunity of ingesting some of that water. And for many communities, bathing in rivers and other forms of water that's been accumulated naturally is what is the facilities that they have available. But then there are also religious activity, and particularly rural poor, but there's also your urban sectors that are at risk of getting that exposure. So some of the efforts has been to attempt to quantify the risk associated with these various activities, but most of it has been on the consumption aspect. Pharmaceutical and freshwater environments, that has been documented, it's been well documented. It's been documented specifically in developed countries. There's a lot of information, but we are lacking behind insofar as the African context is concerned, with some pockets of excellence and information coming through. But the question is always, is what we are producing, is that sufficient? From the previous study that I've just shown now, is that this particular study has shown that maximum concentrations in Africa are nearly 20,000 times higher than in Europe. That sulfur methoxazole, not specifically featuring sulfur methoxazole, was one of the most commonly detected compounds, and later on I'll show some further results on for some findings. But importantly is some of the measured concentrations were up to close to 8,000 times higher than ecotoxicity endpoints, and that was for Africa. So when does a Scopus search for AMR in Africa, that is typically what you are getting, and that was just a Scopus search. Some of the other databases that could be a little bit higher in terms of the actual numbers. But what is important is that we have realized that research will find what the actual data and what the actual information that we have to deal with. And there's this drastic increase over the past 5 to 6 years in terms of the numbers of publications coming through, and that's a positive. A systematic review on antimicrobial use as well as resistance in agriculture and food production systems in Africa. And that is from 1980 through to 2021. What they have found was that there were just more than 470 articles that was available, and after processing all of it, there was only 160 that could be included for the whole of Africa. Globally, there are about 60,000 tons of antimicrobials that are used in plant production and a similar amount in livestock production. The reporting rates for AMUs in Africa as well as AMR is low. It's increasing, which is encouraging, but it's still very low. Also, there were between 27 and 30 countries over the past 5 to 6 years that were reporting antimicrobial use to the WHO as well as the Animal Health Organization. Some of the antibiotics that are used in animal as well as in plant production is tetracycline and some of the other ones, but specifically within animal production is tetracycline, ampicillin, trimethoprim, and sulfamethoxazole combination was very high in terms of when they look at the AMR in Africa. There's a lot of information in this particular table, and that comes from that study. But what I want you to focus on is that some of the antibiotics that I've mentioned, they are the antimicrobial agents, include the tetracyclines. There's also vancomycin and a number of other ones, but tetracycline is coming through basically in all of it. That is from different countries across the spectrum of Africa. But also that virulence genes with the antibiotic resistance, virulence genes were detected, and this is specifically with Enterococcus species. Now, Enterococcus faecium is one of the escape pathogens that we are currently surveilling for in healthcare settings, and we're setting up surveillance programs within the environment as well. But if you look at these virulence genes that were detected, it was just detected, it was there, it is part of the DNA of these isolates. Some of the study, or one of the studies that we have done, where we've looked at antibiotic resistance in Enterococcus species from surface waters, we see similar trends in antibiotic resistance. They are the beta-lactams, ampicillin is there, vancomycin, tetracycline, and this is from surface water. But also that virulence factors associated with those pathogens from the animal production is also prevalent in the surface waters. But importantly is that another study that we've done showed that these virulence genes were not just there as part of the genome, but they were actually actively producing extracellular enzymes. So this shows that there is that definite link between the animal production pathogens and what we find in the environment, and whether that comes directly from them, whether those are the actual pathogens from the environment, that is undetermined. So the antibiotic resistance in the environment is a critical phenomenon, and then something that we have to take very seriously. Antimicrobial resistance in Africa, this systematic review that basically included all the different countries within Africa. That was focusing on data quality, specifically microbial data, demonstrating that the quality of data is a concern. That AMR data was not available for more than 40% of countries in Africa, and that resistance to commonly prescribed antibiotics are quite high. Some studies that we have done, and that was NRF as well as WRC funded studies, have demonstrated that in raw water as well as in treated water, antibiotics resistance microorganisms were isolated. And that's another study from a recently completed WRC that has demonstrated clearly these links between raw and drinking water. Some of the knowledge gaps in the assessment of antimicrobial resistance in surface water, because that is one of the main issues within AMR in the environment, is that what are the gaps? There's been some studies, and I'm highlighting this one, it's a very recent one from 2021, where some of the issues are highlighted, and I'll get back to some of them. One of it is detection of antimicrobial resistance, whether it is within the organisms, whether it's within genes that we find, but also the environmental concentrations of antibiotics and how all of that can be fit into a risk assessment. For a minute, I will then come back to that risk assessment and how far we've come to it. But there's also these other substances that can act as co-resistant selectors. So the authors have looked at the approaches, also what the knowledge gaps are and solutions. So things are being done, and some more research and lessons that we can take from these to ensure that our research agendas would cover these gaps so that we could get the solutions that is required for us locally, regionally, as well as globally. So let me get to some of the solutions. In terms of how do we prioritize policy actions for these chemicals and microbial drinking water risks, those authors have basically looked at what the current scenario is and how we go about doing it. And that is basically to do with concentrations. It is straightforward concentrations and the potential impacts that it could have and how we score these. They've gone on, and I'm not showing the refined approach that is available from their study to have a more global view on risk assessment and what the criteria is that one could be using with more and more data that is becoming available and more streamlining of the approaches that are used. One of the things that we have proposed some time ago is what are genes? What are the resistant genes within drinking water systems? We've also conducted a study for the WRC on that in that regard. But these authors have looked at the prevalence and host-associated antimicrobial strains in disinfected and non-disinfectant drinking water systems. And they've used a metagenomics approach where it is just get the DNA out and then sequence whatever is available. So they found that AMR traits were higher in systems with disinfection, specifically chlorine, but that the AMR is associated with microbial community structure. That was one of the findings from a WRC report as well that has demonstrated this association. They've proposed that absolute concentrations of AMR within these systems and the functionality would be important for risk assessment of AMR. And now very recently, I think this paper was published about a month ago, where for the first time a risk assessment of antibiotic resistant genes were focused on, and they have come up with a potential plan of how to go about that. So taking into account ARBs that were isolated, ARGs that were detected, as well as functionality in the three compartments, that is raw water, the drinking water at the treatment plant and immediately after treatment, as well as in tap water. We've come up with a nice elaborate plan where they have all these evaluations taking place and they are able to give values to it and then use that information to provide the risk assessment, but also for the purpose of use and what potential interventions are needed for that water before it is consumed. So they've looked at known resistance. They've also looked at novel resistance. They've looked at the potential to confer mobile genetic elements the use of antibiotics. So there's a number of different aspects. There's also the chlorine resistance that they've included in this. So a number of what we had proposed previously are important aspects. These guys have basically, they've taken all of that and they've put it into a risk assessment model. Then an aspect of AMR removal efficiencies. And we know that normal wastewater treatment plants were, the conventional plants were not designed to remove these chemicals, particularly the chemicals of emerging concern, including the antibiotics. But there are other processes that could potentially be employed. And these researchers, Hiller et al., have looked at what the literature says about the potential of removing AMR to levels that you'll find in the environment, background levels that you will find in the environment. And they've demonstrated that advanced wastewater treatment processes are available. Costly, but they are available to have that removal potential. Then there's also the question of alternatives to antibiotics. This paper by Allen et al. Has also done a literature review and looked at what are the alternatives to antibiotic use, and vaccines is definitely a pop, expensive in some cases, and then that is for human as well as animal production, but it's there. And there's a number of other ones included in it. As phage therapy, that's endo and exolysins, bacteriocins, and predatory bacteria. And I know that there are some groups within South Africa that's working on those specific approaches, specifically within the microbiology at Mafeking. There is work on phage therapy, on potential applications of that. But then there's also alternatives to antibiotics coming from nature. One of the, or two of the sessions, session 2 and 3, will specifically focus on some of these alternatives. This is a typical example where the focus was not just on an antibiotic alternative, but also to study and to look at whether it is contributing to antibiotic-resistant genes dissemination, and not just an alternative. So it is very encouraging that this is happening globally, but specifically within South Africa and the greater Africa as well, providing some solutions. There's stewardship. There's also the role of civil society organizations within our approaches, and tapping into that, ensuring that we get our civil society organizations as part of our effort. From that last study on civil society organizations, this is basically what they came up with, and this is for the national AMR action plans. In green is where those are in place and government has okayed it, but there are places where it is being developed as well as where there is no information on AMR action plans available, in red as well as in the grey ones without the dots. So what is in World Antibiotic Awareness Week or Antimicrobial Awareness Week? It's more than just a call for us to be aware of it. Also, it is more than just a call from the WHO using that slogan, but is truly to become champions for it. The WHO is also currently, there's a lot of activities that are happening and specifically today is this implementation and financing of AMR national plans that are being discussed. There's also conversation on antifungal or fungal infections and AMR, and a number of other initiatives that are taking place. From specifically South Africa and Africa, and with our the differences in how our water is distributed, there's definitely this need for risk assessment of AMR in aquatic environments. Understanding the background levels of antimicrobial substances is important. Also the effects of these low levels of antimicrobial substances that we may find, but specifically under local conditions. And also the physical conditions locally, as well as the functional trait. And to have that data work for us within risk assessment models that are being generated. So some of the take-home messages is we need to reflect, as researchers, reflect on work that is being done within antimicrobial use as well as antimicrobial resistance, and reflect on what do we really have and take good stock of that. But also, what do we need for impact? And that is what we need, what we have to prevent. If that number is definitely going to increase, the number of deaths within Africa. But from the interventions that took place within the COVID environment, I foresee that with our concerted efforts and with enhancing our efforts, we will definitely be able to curb the spread of AMR within Africa and avert that disaster. We know that antibiotics and the antimicrobial substances in water originate from the healthcare settings, agricultural return flows, but also from urban settings. And it will land up in our drinking water as well as our irrigation water and animal watering that is available. And in some cases, definitely on-site treatment options should be part of what we advocate for some of the high-risk areas with some implementation. And also the embracing new technologies, nanotechnologies, for example, the advanced oxidation processes, etc., that can remove these ARTs. Then I have a number of aspects in terms of the messages, but the surveillance of AMR in the environment, I think that is critical. Well-organized funding opportunities to perform the basic research, to get the data, but also funding opportunities for strategic impacts locally, regionally, as well as globally. Funding opportunities to basically look at how our national action plans fit in with our basic research and what basic research we are performing to provide sufficient information for national action plans to be put in place. And how the research will also affect our policies to combat and the implementation of this One Health approach and hygiene practices, biosecurity in animal production, improved vaccination regimes, etc. So thank you very much. That is, thank you to the organizers and thank you to the team that has listened. Thank you.
Good morning, ladies and gentlemen. I'm Deirdre van Wyk, and I'm going to be chairing the first session. Thank you, Prof. Carlos, for your very enlightening presentation on the topic at hand. Just a few housekeeping rules for speakers. Each speaker has 15 minutes, and we're only going to take questions after the four speakers have spoken, and then there will be 10 minutes for questions and answers, and then we will break out for 15-minute break. So with further ado, the first speaker is Ms. Raisa Biko, and she will be enlightening us on the characterization of Enterobacteriaceae from microplastics in a microcosm setting. Over to you, Raisa.
Okay. Hi, everyone. Can everyone see my screen?
Yes, we can, Raisa.
Okay, that's perfect. Good day, ladies and gentlemen. My name is Raisa Biko, and my research is based on the dispersal potential of microplastic-associated coliform bacteria and antibiotic-resistant genes in the ocean. This is a microcosm study. My research is supervised by Prof. Carlos Pezeiro, Dr. Lesego Molale Tom, Dr. Charlotte Mini, Prof. Henk Bowman, and Dr. Odd-Gunnar Bakmark in Norway. South Africa is surrounded by a 3,100-kilometer coastline that is rich in biodiversity and natural resources. However, this fragile environment is under threat by urbanization, agriculture, and general marine pollution. The coastal areas of Southern Africa are home to 14 marine sewage plant outfalls, some pumping an estimate of 44 million liters of wastewater into the ocean each day. This poses a hazard as wastewater contains a variety of chemicals and microorganisms that may affect the ecological health of the marine environment and can also pose a threat to human health. Studies show that microplastics are present in the effluent of wastewater treatment facilities decanting into the ocean of South Africa. Through indiscriminate disposal of plastic, up to 10% of the produced plastic ends up in the ocean, with polyethylene, polypropylene, and polystyrene being the most prevalent. So this brings me to my research question. Does microplastics, as one of the world's emerging pollutants, have the ability to accelerate global antibiotic resistance? The aim of this research study is to isolate E. coli from microplastics in a simulated marine sewage plant outfall, with the objectives as to set up a microcosm simulating the environmental conditions of sewage outfalls in the ocean, to isolate E. coli in biofilms formed in the microplastics, and to characterize whether the isolated E. coli contain antibiotic-resistant genes. So this would give us an idea as to whether these pathogens would have a potential to colonize, survive, and disperse by means of microplastics in the ocean. Firstly, I would like to highlight the importance of a microcosm study. Microcosms are used to simulate and predict the behavior of natural ecosystems under controlled conditions. This will then allow us to make a comparison of the actual behavior of these microbiomes in the environment. So a microcosm has been set up using virgin polyethylene, polypropylene, and polystyrene beads, which are about 2 to 5 millimeters in size. It has undergone sandpapering and UV radiation to simulate natural degradation. These plastics were then added to a ratio of wastewater and salt water, simulating a perimeter near the outfall. It was then left for 15 days for biofilm formation and then observed using scanning electron microscopy. So the SEM results have shown biofilm formation at day 2, and it has increased on all 3 of the plastics as time went on. So biofilms can easily be seen at day 8. A positive control for the SEM was E. coli 80cc broth in the same conditions to observe biofilm formation and how they attach to plastic. So a clear biofilm can be seen on polypropylene at day 15, as that's where most of the pathogenic strains originate from. Please bear in mind that this image of polypropylene at day 15 is an experimental image. Table 1 shows a maximum number of isolates that were obtained using biofilm removal methods. Coliforms were isolated on mLg agar, thereafter gram stained to ensure purity. Most of the coliforms were isolated from polypropylene particles as previously stated. These coliforms also have the ability to produce extracellular enzymes such as proteinase, lipase, DNase, and hemolysin. Table 2 shows the emergence of antibiotic resistance. The Kirby-Bauer disk diffusion method has been used, and the coliform species showed a high percentage of resistance to amoxicillin, oxytetracycline, ampicillin, and nalidixic acid. DNA extraction, 16S rRNA PCRs, and sequencing was done on the isolates, and the isolated strains can be shown with a red dot. The results showed a strain of Shigella flexneri. It falls into the same grouping as most of the strains with the genus Escherichia. Using BLAST identification from the 11 obtained sequences, 5 isolates were then shown to be Escherichia coli, 4 isolates were Shigella flexneri, and 2 isolates were Enterobacter species. Table 3 shows the genotypic and phenotypic characteristics of the identified coliforms. The results showed a coliform strain of Escherichia coli resistant to 8 out of the 10 antibiotics tested for. And these antibiotics are most commonly used to treat bacterial infections in a clinical setting. The Shigella flexneri strain identified favor proteinase and lipase as the enzyme of choice. The Escherichia coli strain that was identified showed pathogenicity as well as excretion of extracellular enzymes. DNase activity was witnessed in 2 coliform species. The housekeeping genes UIDA, MDH, and LAC-Z were present in most of the coliform species screened for. The MDH gene was present in 10 out of the 11 identified isolates, as shown in Table 3 previously. Polypropylene has been found to harbor the most pathogens, with 37% of these isolates being resistant to 8 of the antibiotics tested. The sequencing results shown in Figure 10 show the presence of Shigella species. The 16S rRNA gene fragments are not suitable to differentiate between E. coli and Shigella species due to a close relatedness. Even so, Shigella species have a higher pathogenicity with a low infectious dose, making it of utmost importance to correctly identify these microorganisms. The housekeeping genes of E. coli are highly conserved, but they were found in Shigella species as well. E. coli is a fecal indicator organism, so it must be found in sewage. So it's expected to be found in the outfalls as well. But from a fresh water, an increase in salinity may be an environmental stressor, thus putting these microorganisms in a viable but non-culturable state. According to a study, there's a seasonal variation in the length of time that E. coli is ineffective in seawater, as it is less viable in summer than in winter. Shigella flexneri employs multiple mechanisms to survive high salinities, as it also survives in bile salts. So many of the coliform bacteria were resistant to multiple of the antibiotics tested for, and many of these isolates can be linked to biofilms on polypropylene. All the coliform isolates show the presence of housekeeping genes, and this is a cause for concern as these isolates use microplastic-associated biofilms as a survival mechanism, which may allow it the ability to disperse through the ocean. It could pose a public health risk towards the immunocompromised individuals as well. Thank you. I'd like to acknowledge the Noordwes-Universiteit, Kennock Institute for Biosafety, the National Research Foundation, and this project forms part of the Sun Oceans project. Thank you.
Thank you, Ms. Biko, for your lovely presentation. Our next speaker is Mr. John Oluketu from the University of Johannesburg, and he will be enlightening us on the diversity and antibiotic resistance profile on diarrhea causing Escherichia coli from the environmental water. Thank you, Mr. John.
Thank you. Thank you, Chair, and thank you. Hi, everyone. I don't know if you can hear me. Okay. Okay. It's fine now. Okay. Fine now. Yes, I will be presenting a part of my. Is it fine now? Okay. I'll be presenting a part of my research project, and the title is the diversity and the antimicrobial and body resistance profile of the regionic E. coli isolated from environmental water in Johannesburg, South Africa. And this study is was under the supervision of Dr. Atisha Singh and Prof. Bernard from Water and Health Research Center, University of Johannesburg. Just a small background. Antimicrobial resistance has become a global burden, that as most of my previous presenters have described. And this is like a new threat worldwide. And it has been reported that 700 people die annually and globally due to antimicrobial resistance. And this number will rise to 10 million per annum by 2050 if appropriate action are not taken. And the study has demonstrated that antimicrobial resistance has been disseminated in animal, clinical, and environmental settings. This emphasizes the importance of One Health approach in fighting antimicrobial resistance. Study also have reported a large volume of antimicrobial resistant bacteria in aquatic environments. And the bacteria E. coli is widely recognized due to its role in spreading antibiotic resistance in water and environments. And study also described that several antibiotic resistance mechanisms in E. coli isolates, including conjugation and the restored gene transfer. So, understanding the different ways that antimicrobial resistance genes spread and their transmission between various components of the ecosystem will help to prevent antimicrobial resistance. And currently, there is no strategy in place to contain and track the movement of antimicrobial resistance, because they show that most of the study is based on clinical setting. And in South Africa, only few data are available on virulence and antibiotic resistance profile of bacterial E. coli. So this pushed me on the aim of my study was to evaluate the antimicrobial resistance pattern of the regionic E. coli isolates from the environmental water, and also to characterize the antibiotic resistant gene, especially in Johannesburg, South Africa. So this is like a big picture of my material and method what I was doing with my study. So I was collecting sample from rivers and some well, and all was in the 11 sites. So somewhere this small figure here, you can see, it's where we are collecting our sample in Cliff Rivers, Jackskey, and the Enops, in Centurion, somewhere in Ellis Park, and in Ekerhof. So what we did with the sample, we did filtration technique, and we subculture in selective media. So we use high chrome selective media, and the presentive isolates we confirm using Vitek. And also we did antimicrobial susceptibility, use also Vitek automated system. Then our E. coli preventive, we store them in glycerol. So at least we have a total of 170 samples that we extracted DNA. So from that extracted DNA, we use different technique to detect virulence genes. So we use 11-plex PCR for virulence genes, and we also use for antibiotic resistance testing. We targeted ESBL producing E. coli. Also, coalition resistance gene is a part of our study that we haven't reported yet, but I just put in this big picture. And at the end, we will do genotyping with whole genome sequencing, especially from the hybrid pathotype that we detected in our study. And at the end, we report, we did analysis and report in the form of publications. So this picture, this image just shows our the gel, what really we take from our study. Using multiplex PCR, we found that 60% of our isolates were pathogenic or dirogenic, and 40 were commensal. So here, on the right side, there are genes that we are detecting. The MGH was our housekeeping genes, and we have all other genes that were present in our isolates. So from the virulence study, we managed to publish this paper, and it was published last month. And the second part of our study, that is the antibiotic that we are reporting now. So we found that using Vitek, almost 100% of our isolates were resistant to kefiroxil. And if you can see the numbers, so it's 100% resistance, 96% resistant to ceftazidime, 82% resistant to cefotaxime, and all those results show that this is a very big concern. Seeing these results. So this is just the image, the figure that what I was talking about here. If you can see, if you start at the bottom side, you can see that this is a very concern because most of our isolates were resistant to the commonest antibiotic that you use in our area. And if you see if you check on top there, the number 2, the colistin. So we found that 4 isolates were resistant to colistin, but we still need to do further investigation to detect, to target some MCR genes, and that will be a part of our next study. So we run another PCR to try to detect those antibiotic resistant genes. So we started by this ESBL producing, and we target firstly the TEM beta lactamase gene, and it was detected in 42% of our isolates. And we still need to detect that to target other genes such as CTXM for ESBL, and like I say, we're also going to target some MCR genes, MCR 1 to 5, and see if some of our isolates might have the colistin resistance gene. And so to complete this, my study, I will say that this study highlighted the widespread of occurrence of antibiotic resistance trend in aquatic ecosystem of Johannesburg, South Africa. And like I said, this study detected ESBL producing genes, BLE-TEM, and which was 42% of isolate, and this is a concern. So there is a need for studies in South Africa and environment to address antibiotic resistance by applying One Health approach. The implementation of environmental surveillance plays an important role in reducing the potential public health risks to close human contact as well to consumer of untreated water. So I would like to acknowledge all the staff of WHRC and the Global Excellence GES Scholarship, which found helped me to help me with some funding. Also, I would like to acknowledge the Water Research Commission and the University of Johannesburg for funding this project. This is my reference, and thank you.
Numbers of the antibiotic resistance it's really alarming. It's really alarming. Thank you for keeping to the time. We're moving to the next speaker. She is Ms. Sulies van Zyl. She's from the Noordwes-Universiteit, and she will be presenting the prevalence of antibiotic-resistant Clostridium species in wetland systems after the sewage outfall. Ms. Sulies.
Hi, can everyone see me?
Yes, we can see you, we can hear you. We do not see your presentation.
Okay. Alrighty. Is it good?
It's present, yes.
Okay, so hi everyone, my name is Sulies van Zyl and my study includes the prevalence of antibiotic-resistant Clostridium species in a wetland system after sewage outfall. And I'm part of the Noordwes-Universiteit, and this study was done under the supervision of Prof. Jacobus Seidenhout, Dr. Charlotte Mini, and Mr. Ruan Fourie. So firstly, let's talk about Clostridium. Clostridium is known to be gram-positive anaerobic pathogenic organisms that are very stable in the environment due to their spore-forming abilities, and they are known to cause a number of diseases including food poisoning, diarrhea, and also in severe cases, gas gangrene. So wetland systems, if we move to wetland systems, they are of a critical importance for a number of economical and biological benefits due to their ability to trap slow-moving water and then act as natural filters and detoxification by the removal or by removing pathogenic or toxic organisms. They're also known to have a very high nutrient count and unique ecosystems with a large amount of interactions between organisms where horizontal gene transfer is known to be in high degree. This leads us to the problem of antibiotic resistance, since resistance towards an antibiotic is generally known to be accomplished through mutations that occur within the gene of pathogenic organisms. These genes can then be transmitted between different organisms through the process of horizontal gene transfer, and thus will increase the amount of resistant bacteria. It is well known that antibiotics has a large number of uses, and industrial and agricultural runoff is of the main contributors to wastewater being treated. Insufficient treatment of wastewater will result in the presence of these antibiotics as well as a resistant species within the effluent, which will then be subjected into the wetland system. Now, what is the problem? So the most pathogenic organisms that are focused on when looking at the suitability of water is known to be aerobic bacteria. But it is also important to remember that, for example, Clostridium, which is also associated with the intestinal tract of humans as well as animals, and their presence in water is known to be an indication of fecal pollution. And thus, they are, as I stated, known to cause a number of serious infections and diseases. And thus, their presence in the effluent as well as in the wetland system, which eventually leads into the river, is problematic or cause of concern, since they have the potential of infecting a lot of people. And thus, their antibiotic resistance, along with their virulence trait, is definitely needed to be taken, is definitely a need to be taken into consideration when determining the fitness of water. So this leads us to our problem statement that no studies, especially in the Northwest province, has been done or conducted on a wetland system to determine the prevalence of the antibiotic-resistant Clostridium species, as well as their virulent traits, and also the effectiveness of the wetland in actually removing these pathogenic organisms from the environment. So the aim of this study is to determine the amount of antibiotic-resistant Clostridium species within different sections of the wetland, as well as in the effluent, to see if the wetland is indeed functional. And we will also be investigating their virulence properties to see how dangerous they really are. So the aim will be accomplished by a set of objectives, including the isolation of Clostridium species, with the use of membrane filtration, and also determining their multi-drug resistant and resistant profiles along with their virulent characteristics, after which identification will be completed through Sanger sequencing after 16S rRNA amplification. Our method that we will be using will start at sampling, the isolation with membrane filtration, multi-drug and virulence testing through spot inoculation, and then incubating them in anaerobic jars with the use of anaerogen sachets, which will remove the oxygen and produce anaerobic conditions. Phenotypical testing of purification of a certain amount of isolates that will be subject to sequencing will take place. Phenotypical testing will be conducted on them, including gram and endospore staining. The extracted DNA will be amplified through 16S rRNA PCR, and then they will be subjected to Sanger sequencing for identification. So firstly, we look at our sampling sites. At site 1, we started at the effluent of the wastewater treatment plant, second and third location further down the wetland, and the fourth location at the point where the wetland enters into the Mooi River system. So for our results, table 1 shows the number of antibiotic-resistant Clostridium that was obtained via membrane filtration. So the resistance was calculated through the MIC concentrations, which is the minimum inhibitory concentration. So it's the concentration to which a certain organism, in this case specifically Clostridium, is resistant to that specific antibiotic. So it can clearly be seen that the antibiotic greatly decreased the growth or the amount of coliforming units from site 1 to site 4. So there's a definite decrease in pathogenic organisms, which indicates the effective functioning of the wetland system. This table was placed into a graphical visualization. So here we can see again the great decrease from site 1 to 4, which is positive, indicating the effectiveness of the wetland in removing these pathogens. However, ampicillin and tetracycline showed the highest resistance, with tetracycline numbers are off the charts, ranging from 705 isolates from site 1 to 37 in site 4. These are very high in comparison to the other antibiotics. So the reason for this can be tetracycline has over 50 resistant genes that are identified, and it is also one of the most well-known and well-known and most distributed antibiotics used today for these for antibiotic infections. Metronidazole, clindamycin, and chloramphenicol also showed some degree of resistance, but much less than ampicillin and tetracycline. And metronidazole showed the most susceptibility, or the highest amount of susceptibility. And this is supported by studies indicating that the mechanism is one of the most effective against anaerobic, specifically anaerobic organisms. And this is positive since it is still seen that the antibiotic is standing strong against pathogenic organisms. So 25 purified antibiotic resistant isolates were obtained, and from these 25, 23 isolates showed multi-drug resistance. The multi-drug resistant title was given to an isolate when it showed resistance against 3 or more antibiotics. So for the 25 isolates, the phenotypical and virulent traits they were conducted to those tests. And from this, it can be seen that all the isolates tested positive for beta hemolytic activity of hemolysis. And also, all the isolates also tested negative for lipase. So with regards to DNA and proteolytic activity, 30 isolates, or 52%, tested positive for DNase activity. This is concerning since this is higher than the half of the isolates tested positive. And for proteolytic activity, there was some degree of action, but only 7% or 7 isolates, which roughly goes to 28%. Last step was identification, and this was through Sanger sequencing, where we saw that 15 of the 25 isolates were successfully sequenced to species level, and Clostridium perfringens is in the majority with 46% or 7 isolates, followed by Paraclostridium benzyliticum with 4 isolates. This is then Sordalia or Clostridium sordalii with only 2 isolates or 30%, and then also 2 different organisms that aren't Clostridium but are both pathogenic organisms, sulfate-reducing bacteria, and also facultative anaerobic, which entailed them with the ability to grow on the specific growth media as well as in the anaerobic conditions. So the presence of these Clostridium species, as well as the 2 unknown, are of concern, since they are known to cause infections and they show a great deal of virulent traits as well as antibiotic resistance. So Clostridium is one of the most well-known pathogens and is associated with the intestinal tract of animals and humans, causes severe diseases like, or causes illnesses like food poisoning, diarrhea, stomach pains, and so forth, and in severe cases, gas gangrene, and they're also largely present in water. So with regards to benzyliticum, which was the second highest, this is a relatively new strain. It was only introduced in 2016. It shows close resistance to Clostridium bifurmentis. It has been isolated from soil in marine habitats as well as polluted water and some clinical sources, but they are relatively low in the region of pathogenicity. For Sordalii, they are known to follow spontaneous or medically induced abortions and cause uterine infections, arthritis, and peritonitis. So for the Proteus mirabilis and the Rotella planticola, which is the unknown organisms, they're also known to be generally found in the environment, and is also known to cause infections where the mirabilis has high urease activity and causes urinary tract infections or UTIs, and the planticola has a variety of illnesses which include cystitis, pancreatitis, hepatic diseases, and so forth. So all of these are of great concern, since the high antibiotic rate will result in the insufficient treatment of diseases caused by these organisms. In conclusions, we can see that the wetland system was indeed functional in removing these pathogenic organisms. The isolates definitely entailed virulent traits, with all testing positive for beta hemolysis activity, and also half of the isolates for DNase activity. And ampicillin and tetracycline showed high resistance, which is of great concern since these are the most used antibiotics. Excuse me. A further investigation can include the mechanism used within the defense mechanism used by metronidazole within Clostridium species, and this is relatively unknown, as well as investigating the presence of antibiotic-resistant genes within wetlands. And there are also, we can further look along the merger of the wetland system with the river to see how many of the pathogenic organisms are actually transmitted down the river system, and that is actually exposing people to pathogenic risk. And also we can investigate the effect of seasonal variations on the level of these Clostridium species. Thank you so much.
Thank you, Ms. Van Zyl, for your very enlightening presentation. We have a lot to think about. A lot of questions will be coming. We're moving to our last speaker in this first session. It's Mr. Lee Hein from University of Johannesburg. He will be presenting on antimicrobial resistance associated with pathogenic bacteria from the environment in the soul of the Yukskei. Over to you, Mr. Lee.
It's Ms. or Dr.
Ms. Apologies for that.
It's all right, not a problem. Can you see my screen?
Yes, I can.
I'm not going to share video of myself because it does disrupt my internet speed.
No problem.
Cool. Why is this not working? Oh, there we go. So I'm going to be presenting on the antimicrobial resistance associated with pathogenic bacteria that were isolated from environmental water, specifically the Yukskei River. So just some abbreviations. I'm sure most of the audience panelists are familiar with these abbreviations, but just to give you those. Many South Africans still have limited or no access to safe or improved water sources, and therefore the presence of antimicrobial resistant pathogens in environmental waters poses a significant risk to human health. Today, I'll be presenting just on the AST data obtained during a multidisciplinary pilot study which investigated the water quality of the Yukskei River in Johannesburg. The published data included in-depth microbial and chemical analysis of 6 water samples. 2 water samples were collected for each month in June, July, and August in 2018. This was upstream and downstream from Victoria Yards in Lawrenceville, central Johannesburg. And these samples were transported to the Water and Health Research Centre laboratory at 4 degrees Celsius. They were then cultured onto several selective differential media, including DCA, TCBS, and sorbitol mkonki, as well as onto Campylobacter blood-free agar, which was cultured in duplicates both aerobically and microaerophilically. Pure cultures of selected isolates were then grown on Muller-Hinton agar for identification and antimicrobial susceptibility testing using the Vitek 2 compact automated system. Now, in a bit more detail, for the identification of gram-negative bacilli, gram-positive cocci, gram-positive bacilli, and yeasts, GN, GP, BCL, and YST identification cards were used, respectively. For the susceptibility testing of the gram-negative bacilli and the gram-positive cocci, N256 and P645 AST cards were used. The minimum inhibitory concentrations for each antibiotic were interpreted as susceptible, intermediately resistant, or resistant, and thereafter were categorized manually as multi-extensively or pan-drug resistant. So of the escape pathogens, only 1 of these was not detected. Examples of all the rest were detected. The one that wasn't was Enterococcus faecium. In addition, several examples of diarrhea-agent bacteria were identified, along with other clinically significant human pathogens. Specifically with regards to the AST of the gram-positive cocci, 8 were subjected to AST. All of these were identified as multi-drug resistant. That is, they were resistant to antimicrobials in 3 or more antimicrobial categories. None of them, however, was characterized as extensively or pan-drug resistant. With regard to the gram-negative bacilli, 25 were subjected to AST. I've split the tables just so that people can actually see all of the results. So 2 of these isolates were susceptible to all the antimicrobials tested. 5 were resistant to antimicrobials in 1 or 2 categories. 18 of them were classified as multi-drug resistant. 2 isolates, both of them E. coli, were categorized as extensively drug resistant, that is resistant to drugs in 10 out of 12 of the antibiotic classes tested, and none of these isolates were pan-drug resistant. 8 of the gram-negative bacilli were also found to be ESBLs, or extended spectrum beta-lactamase producers. 6 showed high-level AMP-C resistance, and 4 were extended spectrum carbapenemase producers or were impermeable to carbapenems. 2 of our isolates were resistant to colistin. Now, the gold standard is to do broth microdilutions to confirm that result, but this was not done, and it is recommended that in future studies at our unit, we will do the broth microdilutions to confirm any colistin resistance. So the results of the present study correlate with other South African studies, including those involving clinical and environmental isolates. The resistance of 2 of our isolates to colistin is worrying, as this is an antimicrobial of last resort and is reserved for the treatment of multi-drug and extensive drug-resistant infections. Infection with antimicrobial resistant bacteria, as we know, increases morbidity and mortality and places an increased financial burden on the healthcare system, which, as we all know, again, is currently already strained due to the COVID-19 pandemic. And South Africa is a low income and a resource limited country. And as such, the economic costs of antimicrobial resistance will have a greater impact on our country than that experienced by more wealthy nations. And antimicrobial resistance, obviously in the environment and clinical isolates, needs to continue to be monitored so that we can, learn as much as we can and hopefully fight the pandemic of antimicrobial resistance as well. This study was conducted at University of Johannesburg's Water and Health Research Center and was supported by UJ Peets. Thank you very much.
Thank you, Ms. Hein, for your lovely presentation. All of your presentations have the same thing in common, and that is antibiotic resistance or antimicrobial resistance. I would like to ask for questions. Is there any questions? I have one of my own, if I can go first. We have 15 minutes for questions and answers, and then we will break out for a small break. So these numbers of antibiotic resistance and antimicrobial resistance, it's a huge problem. I'm sure later on in other presentations, some of the speakers will come with surveillance programs in other implementations that we could use. But what do you guys think, looking at your own research? What is your opinion? What does these numbers mean for you guys in the environment specifically? These high antimicrobial resistance or high antibiotic resistance in your research? Any one of the speakers can answer.
Perhaps, Dr. Deirdre, if I should then start out with a potential answer to the question that you are putting out. We are seeing these high numbers of resistant organisms in the environment. And your question, if I interpret it correctly, is what do you think? Why is it like that? Why do we find these high numbers in the environment? Is that correct?
Yes, Prof. Carlos, that's correct.
Okay. So for a long time, the focus was on the clinical settings, that is human as well as animal, and managing the clinical settings for AMR. The environment has been neglected until recently. There wasn't much focus on what is happening in the environment. It's now only recently, with a renewed effort to get data of what is happening in the environment, that we are observing these very high numbers. Obviously, there are background levels of some of these antimicrobial agents within microbial communities, within soil and water, and those are survival strategies that microorganisms use. But within these systems, there's also the growth of microorganisms as well as the dying of them. And when they go into that senescence phase, the cell wall basically breaks open, and whatever is inside is then released into the environment, and that includes the DNA, and those DNA would be taken up by organisms within that ecosystem. So we didn't have much data of what is happening in the environment, so that's one of the reasons why all of a sudden when we started looking, we are finding these high levels of antimicrobial resistance amongst the microorganisms. Some of the resistance is also innate, and that is why we have to look at what are the genes and whether those genes are actually expressed when we see a resistance pattern. So resistance should not just be taken as it is a genetic reason why the resistance is occurring. So there's a lot more information that is coming to the fore, and that we are getting, and that we are grappling with. And within the presentation that I've done is I've been asking some of these questions. We need to know what is happening within our systems, and how local conditions can contribute towards antibiotic resistance or antimicrobial resistance in general. So that will be my take on it. Perhaps somebody else can also provide some explanation. Thank you.
Thank you, Prof. Carlos. That answers my question. We have one online question.
Okay, I see John speaking here, Bulukauto. Yes, to answer to this question, like Prof said, so it was like neglected to the studying environment. And I think what I took on my literature review, I think the strategies should be put in place to contain and track the movement of antimicrobial resistance in the environment. That will be one of the key, especially to educate people about the One Health approach in fighting antimicrobial resistance. So I think One Health approach is really taking into consideration and try to train even the healthcare workers in the clinic and the people in the working in the animal settings, in the farms, abattoir. It will be also one of the solutions how to track this antimicrobial resistance in the environment. That was my contribution. Thank you.
Thank you, Mr. John. We have one more question from Lawrence Lang. He wants to know, can we as scientists implement a program, surveillance program, or tools? Which tools can we implement to combat antimicrobial resistance in the environment? From what I do know, working on some of the WRC projects, there are some programs, surveillance programs, that are being implemented to combat certain areas of this antimicrobial resistance. Anybody else who can also contribute to this question? Is there any other questions? Then we will go to a break for 15 minutes, and then the second session will start of the day. Thank you for participating in the first session, and thank you to each and every speaker who kept to the time and who brought a very enlightful and really direct presentation as to what is happening, what is the levels, and now we need to look at what we can do to combat these antimicrobial resistance for the future and for our environment. Thank you.
Thank you.
Welcome back, everybody, and I hope you enjoyed the first session and that you have your drinks and snacks ready for the second session, where we will be delving more into the effect of antimicrobials on the human health specific. At the Agricultural Research Council Biotechnology Platform. After completing his undergraduate degrees in genetics and statistics, he obtained a PhD in bioinformatics from the University of Pretoria. Due to the cross-cutting nature of the ARC biotechnology platform, Rian is involved in numerous projects and research endeavors across the agricultural value chain. These include the surveillance and identification of foodborne pathogens and antibiotic resistance. Currently, a large portion of research is dedicated to metagenomics and the metagenomic analysis of diverse environments. Dr. Pierneef, the floor is all yours.
Good morning, everybody. My name is Rian Pierneef, and I'm a senior researcher in the BTP Core Agricultural Research Council. Thank you for availing yourselves for this virtual symposium and my talk, which is titled Next Generation Sequencing-Based Antimicrobial Resistance Surveillance, the Gen Z of AMR surveillance. This presentation will be focused on the application of NGS and in particular metagenomics in wastewater epidemiology and resistance surveillance. I'd like to start off with some acknowledgements and thank all the individuals and stakeholders who've assisted me in these research endeavors presented here. Some of the work which will be presented today is part of a larger project, unselfishly supported by various collaborators within the Success Network. I'd like to highlight the SAMRC TV platform, and in particular, Dr. Awolane Mchembele and Prof. Marty van der Walt for their valuable contributions and energy. The picture in the bottom left is of Awolane in her natural habitat, sampling as always. Then Dr. Mchunu, who is always ready with an answer or insights and brings a wealth of knowledge and expertise to the table. Last but not least, a big thank you to the WRC for all the support and dedication. Without the WRC, none of this would be possible. So let's have a look at the history of next generation sequencing. Nobel Prize winner Frederick Sanger is generally regarded as a pioneer of sequencing, and Sanger sequencing is till this day widely used. He published his rapid DNA sequencing method in 1977, and in 1981, the sequence and organization of the human mitochondrial genome was published. In 1990, the Human Genome Project formally began, involving numerous countries. This international project was expected to take 15 years. During the 90s, various breakthroughs, such as complete cellular and eukaryote genomes, were published. The early 2000s saw a rough draft of the human genome finished by the Human Genome Project, earlier than anticipated, mainly due to the advances in the genomics field. This was followed by various second-generation sequencing technologies, and in 2008, the Human Microbiome Project was initiated. In 2011, third-generation sequencing technologies were rolled out, which produced much longer segments of sequencing data, or reads. In 2019, the Human Microbiome Project entered its third stage, and this project clearly illustrates the importance of metagenomics and microbiomes. But how does next generation sequencing work? Specimens or samples are collected and pure cultures are grown. Thereafter, DNA is extracted and a library prepared. A library is prepared by fragmenting the DNA sample and ligating it with commercially available adapter molecules. Adapter molecules act in the hybridization of the library fragments to the matrix. Moreover, adapter molecules provide a priming site. The library is converted into single-stranded molecules, and then amplification creates clusters of DNA molecules. Each cluster acts as an individual reaction where sequencing, or called a run, is performed. At the end of the reaction, each NGS run provides a large amount of raw data. This data can be analyzed using a variety of available bioinformatic tools. This is all a bit technical and boring, is it not? Next generation sequencing is very similar to building a puzzle. We start off with our favorite picture and surgically cut it into thousands of pieces. Then the fun starts, when we try and put all these pieces back together. Now, this can be done using two methods. The first is a reference-based method, where one would look at a copy of the picture and try and place all the pieces together using copy as a reference. The other would be a blind approach, where edges of the pieces would guide us as to where a piece should be placed. Once we have finished putting all these pieces back together, it's possible to look at the picture as a whole and make various scientific inferences. The completed picture would be able to inform us with regards to where the picture is from, which era it was painted in, if it's valuable or common, and much more. In essence, by completing the genomic puzzle, we have a better picture of the capabilities and potential of an organism of interest. Applications of NGS include metabarcoding and metagenomics. Metagenomics is the study of genetic material recovered directly from environmental samples and has been defined as the application of modern genomics techniques without the need for isolation and lab cultivation of individual species. This is great, as we do not have to complete the laborious and time-consuming protocols involved in the isolation and cultivation of individual species. Metabarcoding and metagenomics can thus be thought of as a large genomic puzzle that details all the microorganisms present in a sample. Building this puzzle piece by piece provides us with a holistic picture of all the little entities and the relative abundancy with which they occur in an environment. We are further able to compare different puzzles with each other to see which microorganisms are present or absent in each puzzle. Now on to the most important topic of the day, antimicrobials and antibiotics. The term antibiotic was coined in 1942 by a microbiologist named Salman Boxman, who defined it as inhibiting the growth or metabolic activities of bacteria and other microorganisms by a chemical substance of microbial origin, and have been used by humans for an extended period of time. Tetracycline, which is absorbed into the bone, has been found in ancient skeletons from Sudan and Egypt, suggesting that these populations consumed antibiotics in some form, either knowingly or unknowingly. The early 20th century saw a wealth of antibiotic research and compound discovery. The rapid rate of antibiotic discovery and their unprecedented success at treating a plethora of diseases led to the overly optimistic view that infectious diseases would soon be entirely conquered. Although some experts were skeptical, in 1967, it was declared that the war against infectious disease had been won. This optimism would be shattered by the rise of antibiotic resistance, with even Alexander Fleming voicing concerns of the potential danger of antibiotic-resistant pathogens in his Nobel Prize lecture. We are currently in the precocious situation where bacteria and other microorganisms are rapidly evolving, and there is a substantial decrease in the rate at which new antibiotics are introduced. Similar to the tortoise and the hare, and unfortunately humans are the ones with long floppy ears. In essence, we are losing the arms race and need to act rapidly. Now, my research efforts are focused on resistance surveillance in various environments. I do this by using metagenomics. As illustrated earlier, metagenomics is similar to building a puzzle. Because we are sequencing everything in a sample, we are able to really focus on specific traits and genomic potential. Metagenomics therefore allows us to detect the resistance potential of the organism in an environment based on the genomic information contained therein. This provides us with another layer to the puzzle, as we can now see which organisms are present and what they are capable of. Now, we recently used this method to do surveillance on wastewater samples from the Tshwane district in South Africa. The samples represented the east, central, and northwest portions of Tshwane and were analyzed using metatranscriptomics, which in essence means we use all of the RNA in a sample to build our puzzles. The samples were collected from Rietgat, Daspoort, and Baviaanspoort wastewater treatment plants from August 2020 to April 2021. After RNA extraction, ribodepletion was used to remove abundant RNAs, such as rRNAs and globin RNAs, which then enables us to focus on the high-value informative portions contained within the mRNA. Now, this step is critical as these samples contain a high portion of host or human contamination, which dilutes the contributions from others within a sample. By doing this, we lower the sequencing cost and increase the resolution of the data. So in total, we generated roughly 100 gigs of raw sequencing data with nearly 20 million paired end reads per sample. After quality control and an additional step to remove any remaining human contamination, we were left with about 18.5 million paired end reads per sample. Now that's a lot of puzzle pieces to put together. As I said earlier, we build our puzzle using two approaches. One is a reference-based approach where we align our reads or puzzle pieces against references. And the other is to use the edges of our puzzle pieces or reads to join the pieces together. The taxonomic results for the samples were obtained using a reference-based approach. For each sample, all the quality filtered, human decontaminated sequencing reads were aligned against the archaeal, bacterial, and viral portion of the NCBI NR database. This allows us to determine the taxonomic composition of each sample at various taxonomic ranks and calculate the relative abundance of each taxonomic grouping. Now, this figure illustrates the taxonomic composition at the kingdom level for each sample. As expected, the largest contributor to each sample was bacteria, which is represented in red. Archaea in green and viruses in blue were also detected and interestingly differed in the relative abundance between the samples. We are further able to separate the data based on the kingdom assignment and compare the taxonomic distributions between samples. Although archaea are not generally regarded as a health concern, they should not be ignored. Archaeal populations are able to assist in the breakdown of organic material and play an important role in converting pollutants into environmental friendly materials. Now, this figure illustrates the relative abundance of archaeal genera in each sample. The large differences in archaeal genera between samples is clearly evident. And a total of 118 different archaeal genera were detected, and each one is represented by a different color. Now, two things to perhaps highlight, other than the differences, are the numerous candidatus genera and the high level of methanogens. The term candidatus indicates that the genera is well characterized, but yet uncultured. Now, this is of interest, and it clearly illustrates the power of metagenomic sequencing and the ability to classify unculturable or yet uncultured genera in a sample. Methanogens are widely used to treat wastewater, as well as organic pollutants. Now, the bacterial portion again displayed vast differences between the samples. This figure illustrates the top 10 genera per sample. With the vast amount of bacteria detected per sample, necessitates that we only look at the top 10. Otherwise, this legend would stretch across numerous pages. Now, of interest was the high relative abundance of genera and generally associated with disease or pathogenicity. Now, this included genera Eremonas, Archobacter, Coxiella, Klebsiella, Listeria, Moraxella, and Pseudomonas, to name a few. Another discussion point would be the absence of E. coli under the top 10, E. coli was detected, but perhaps the time has come to reassess E. coli as a fecal indicator and consider alternatives such as crash phage or other human viral fecal indicators. Now, the samples again displayed a varying viral composition. This figure details the different viral realms per sample, with the most prominent being diploidNA in red and riboviria in light blue. DiploidNA includes tailed bacteriophages, which infects prokaryotes, and herpes viruses, which infect animals and humans. Tailed bacteriophages are important because they recycle nutrients in organic material from their hosts, and herpes viruses are associated with a variety of diseases in animals and humans. Now, riboviria is a realm of viruses that includes all viruses that use homologous RNA-dependent polymerase for replication. Many of the most widely known viral diseases are caused by viruses in riboviria. This includes coronaviruses, Ebola virus, HIV, and influenza. Many reverse transcribing viruses notably become integrated into the genome of their host as part of their replication cycle. As a result of that, it's estimated that about 7 to 8% of the human genome originates from these viruses. Now, the taxonomic puzzles were built using a reference-based approach. Now we move on to another puzzle building approach. Here, we used the edge building approach to construct long, reliable transcripts with high expression. This figure is based on the number of de novo assembled transcripts per sample, with each sample represented by a different color. There's clearly a large difference in the number of transcripts between the samples. Now, this is really interesting, as the number of transcripts are a proxy for the general level of expression or activity in a sample. Some samples seem much more active or busy than others when looking at the number of transcripts per sample. Now, being a scientist, I immediately wanted to know why. Why would one sample have such an abundance of transcripts and another sample from a seemingly similar environment have so much less? Now, this may be due to the amount of sequencing data generated, which is a technology factor. The more we sequence, the more information we get. And as such, this may lead to better de novo assemblies and more transcripts. Or this may be due to a biological factor. Something in the environment or contributing environment to wastewater is causing this increase in activity. Here, we tested some correlations to see what could be the possible reason for the discrepancy in number of transcripts between the samples. In both figures, the number of transcripts on the y-axis. In the figure on the left, we use the amount of data generated as the x-axis or predictor variable. We can clearly see that the amount of data generated, as indicated by the number of paired end reads, is actually negatively correlated with the number of transcripts for this batch of samples. On the right, we have the 7-day average COVID-19 cases as the x-axis or predictor variable. On this plot, it seems like an increase in the 7-day average COVID-19 cases leads to an increase in the transcriptomic activity of a sample. But this may be due to various other factors, and we should be careful of inferences based on guilt by association. The de novo assembled transcripts were aligned against the Comprehensive Antibiotic Resistance Database, or CARD, to identify antibiotic resistance genes, or ARGs. Using CARD as a reference database, we are able to annotate transcripts as possible ARGs according to various annotation levels, such as ARG family, class, and resistance mechanism. This balloon plot indicates the samples on the x-axis and detected ARG families on the y-axis. The size and the color of the circles indicate the frequency. In plots like these, trends across the horizontal and vertical of interest. A horizontal trend or family frequently detected across all samples was the GES beta-lactamase family and members of other antibiotic efflux pumps. Vertical lines or trends were seen for various samples from Rietgat, and they indicated a high ARG diversity. Now, upset plots like these are similar to Venn diagrams, but they're certainly more practical when looking at numerous samples or intersections. The set size of drug classes for each sample is indicated on the bottom left, while the intersection size is displayed on the top. The dots and lines in the center panel indicate members of each intersection. That peculiar Rietgat sample again surfaces as seen by the lone dot in the bottom left, which indicates that this sample has 6 drug classes not found in any of the other samples. Now we can further narrow down our focus on the ARG resistance mechanisms within each sample. Here, each color represents a different ARG resistance mechanism. The differences between samples, represented on the x-axis, is clearly evident. Some samples only have 2 colors or resistance mechanisms, while others have 5. This is a pretty big difference when you take into account that there are only 6 resistance mechanisms described in the CARD database. Now, on a side note, we also had a look at the presence of SARS-CoV-2 in these metagenomic RNA samples, as they were from wastewater treatment plants and very likely contained traces or portions of the SARS-CoV-2 genome. We were able to extract reads mapping to the SARS-CoV-2 reference from 5 of the samples. Now, this is pretty cool, as these samples are highly diverse and there was no amplification or targeting done when sequenced. So in reality, a 30 kilobase genome is competing with various 5 megabase genomes for a spot to be sequenced. Again, the question was asked if the amount of data generated was correlated to the detection of SARS-CoV-2. In the bottom left, it was actually found that the 7-day average COVID-19 cases were the biggest contributor to the detection of SARS-CoV-2 in a sample. Now, this makes sense, as the higher the number of cases in the community, the higher the subsequent viral load in the wastewater samples. So in summary, as you will probably hear throughout the symposium, we are in a very, very bad spot with regards to antimicrobials and resistance, and this requires our immediate attention. NGS and metagenomics is an eloquent alternative to traditional surveillance methods and allows for numerous analysis based on data from a single run. In all likelihood, the time has come to assess the current fecal indicators and align this with work being done on the human gut microbiome. The next step with the data presented will be to investigate seasonal and periodic trends and the possibility that some levels may persist. Now, studies such as these produce a wealth of data which can be used by other researchers for various research endeavors to answer critical questions. Perhaps then to end off with, hindsight is an exact science, and SARS-CoV-2 is a good example of where we have been warned but failed to listen. Let us hope the same does not continue to happen with antimicrobials, as COVID-19 will be a walk in the park compared to an AMR pandemic. So spread awareness and stop resistance. Thank you for your time.
Thank you, Dr. Pierre, for that very interesting presentation. And I'm glad to hear I'm not the only one that uses terms like it's pretty cool or we're having fun with data. So I don't feel so isolated anymore. So thanks for that. Professor Carlos Pesano, so just so everybody knows before I go to questions, Dr. Pierre has to leave shortly after this presentation. So we're going to ask you to ask your questions to him now so that he can leave. Prof. Carlos, you have a question for him?
Thank you, Prof. Pierre. Dr. Pierre, about the sequencing, this was a full metagenomic transcriptomic analysis. And from the data that you have now generated, how applicable is the methodology currently, and how applicable will it be going forward if one would invest in this approach? it is not a cheap exercise, and investing in the approach. Can you make some comments on those questions for us, please?
Perfect. Yep. Thank you, Prof. That's a great comment and question. So what we've seen is that the more technology is used, the cheaper it gets. And this is similar in the high throughput sequencing environment, where it is expensive, but it is getting cheaper. So in general, an approach like this would be probably a good way to scan samples. We're not saying we've got to sequence every sample, but batches of sample, certain periods, certain time intervals, we sequence them. And if we do pick up anything of interest, then we can go back to the sample and perhaps use another method, or then sequence more of the samples of interest. Because yes, agreed, it is unfortunately not feasible to sequence every sample, but in general, if you could at least do one or two of them to get an idea of what the overall population within the sample.
Thank you.
Do we have any other questions? Dr. Yunus.
Morning, Dr. Rian. How are you? Thank you so much for the presentation. I think just from an interest point of view in building up skills for this type of work, because obviously this type of work is going to be important for future wastewater monitoring and for early warning for pandemics. And I have been asked that question before, and I wanted to know, with your opinion, what kind of skill sets would our students, our researchers need right now going forward to make this kind of research more applicable to a wider group of stakeholders?
Great. And that's probably the biggest issue we've got is not the technology, but the human capacity, the ability to analyze these vast amounts of data sets. So skill set is, I became a bioinformatician only after going into postgraduate studies, is you don't need to be a computer scientist or data analyst, but you just need the hunger to test and to play with your data and work with your data. In general, the biggest skill, I think, is data visualization, is to get people to learn how to adequately take a large amount of data and compress it into a single picture, which is then easy to interpret and to show to other people and demonstrate what's been done. And general statistics, maybe just some data analyst work. But yeah, at the end of the day, it's just about sitting down behind a computer and playing with programs and tools that you become more familiar with them and you're more confident in using them.
Thank you. Do we have any other questions from the floor? I'm not seeing any hands. So Dr. Pierneef, I think you're off the hook for today. Good luck with the rest of your day. And thank you for the really interesting presentation you did. We're now going over to Richard Betek. He is from the Noordwes-Universiteit, and he's going to be talking about the synthesis of small molecules as potential antibacterial and antiparasitic agents. There you go. Enjoy.
Good morning, everyone, and hope you can hear me. My name is Richard Mbibete. So I will be taking you on some of the work that we're doing on synthesis of some organic molecules with the hope of countering the effect of antimicrobial resistance in infectious diseases. So just a brief introduction. Infectious diseases are caused by microorganisms that infect other organisms. And there are over 10 million infectious diseases cases reported every year. And there is just high prevalence of these diseases, especially in the African community. And at the moment, the biggest challenge with infectious disease is that of resistance to traditional chemotherapy or traditional therapy for them. So in that, most of the tool to develop or to counter infectious diseases have always been small molecules. And besides treating and prevention of infectious diseases, small molecules are also used to study the biology of infectious diseases. For example, if you discover an inhibitor for a particular protein, you automatically get to know the function of that protein or the enzymatic pathway that is involved. So there are just several ways that you use small molecules to study agents that cause diseases. I will just touch on a few of the infectious diseases that we currently working on or trying to discover some organic molecules on, and I will start with cryptococcosis. This is a fungal disease that is caused by Cryptococcus neoformate. This disease is highly prevalent in immunocompromised people, and is always reported that more than 2 or so million people are affected globally every year, and more than 600,000 people die from these diseases. And like most other infectious diseases, the high burden is in Africa, even though we barely get enough data from it. But the little data that you get shows that Africa is mostly affected. The biggest challenge with Cryptococcus is that there are just 3 drugs available to treat this dangerous disease. And these drugs, one, which is commonly available in the African continent, fluconazole, is not very effective. And the most effective treatment is always amphotericin B and cytotoxin in combination. But this combination treatment is a little bit toxic and is also very costly for the average African to afford. Besides this limited treatment, there are just very little drugs or other compounds in the experimental pipeline to treat this condition. So there is just need for people to get involved in trying to identify new compounds that can be used to treat this disease. So in our bid to find treatment for this disease, we currently looking at piperazine carboxamide as a new class of compound to treat diseases caused by Cryptococcus neoformate. So here you will see we're doing more of the synthesis. So without going too much on the chemistry there, we're just showing you the general structure. This synthesis was done by PhD candidate Claudia Angola. So we start with simple molecules like amine, then do our chemical transformation and the rest to generate several series of compounds that we get to evaluate them. And the evaluation starts by doing single point inhibition assay at 32 microgram per mil. And if any compound shows greater than 70% inhibition at this high concentration, is considered for further dose response inhibition to see if they can generate IC50. So here we use the reference strain to screen for anti-cryptococcal activity, and for compounds that are having activity below 16 microgram per mil, they are considered hit. And for that biological evaluation, especially against the kidney cell line or hemolysis against red blood cells, are evaluated to show that these compounds probably are effective. So after synthesizing the compounds, we send them for evaluation. The evaluation is done in Australia by a charity group there called Community for Open Antimicrobial Drug Discovery. So what we've realized so far is that we've identified a new class of compound that is active against the causal agent of cryptococcosis. And we could generate a good structural activity relationship within this series that correlates so well, and it can help in further exploration or to optimize the activity of this series against this particular pathogen. First, we realized that all compounds carrying a carboxylic acid around position 3 of the quinolone scaffold, they showed some moderate activity, but they are high. They also have strong activity, hemolytic activity. So, for example, if you look at Ka019, Ka016, and Ka013, you see that they have activity from 32 microgram per ml to up to 4 microgram per ml. 4 microgram per ml is really potent activity that is almost comparable with standard drugs in fluconazole in the market. But when you look at their activity on red blood cells, they are also having stronger activity on the red blood cells than the fungi itself. For example, if you look at the Ka016, which shows 4 microgram per ml against the fungi, the activity on red blood cells is 1.7, so it's strongly hemolytic, so they're breaking down red blood cells. So they're not really ideal as compounds for further treatment. But on the other series, when you replace the carboxylic acid with highly lipophilic amides, for example, when you look at the compound Ka018 up to Ka031, you realize that this compound shows a broad range of activity from 32 microgram per ml to almost 8 microgram per ml, which is good activity. And they also lack activity against the kidney cells, and they also do not show any hemolytic activity against human red blood cells. And the most important, interesting compound here is Ka028, which has epiperazine moiety. This compound shows activity against Cryptococcus neoformans of as low as 8 microgram per ml, and has no activity against kidney cells and has no activity against human red blood cells, which shows a potential here. There is great potential to develop this compound to become a good lead compound for Cryptococcus neoformans. And the most important thing here is that this compound class has never been reported in the literature for treatment of this disease, which means you can easily develop it and you will be active even against drug-resistant Cryptococcus neoformans. Another target that we busy with is sleeping sickness. And sleeping sickness affects both humans and animals. In animals, sleeping sickness is referred to as nagana. So in humans, sleeping sickness is also referred to as human African trypanosomiasis. This disease is transmitted through the bite of an infected tsetse fly. It's only common in Africa and in mostly remote areas, and is often ascribed the name neglected disease. This is because you don't have a lot of people interested in to eat, because the people the disease affect are poor to afford any innovations or inventions aimed at this disease. So there is just little research aimed at such disease, neglected disease. So sleeping sickness takes two forms. You have the early stage, that wherein the parasites are located within the bloodstream or lymphatic system, and you have the late stage when the parasite cross the blood-brain barrier and affect the brain, where you have sleep disorder. Generally, the disease has just few drugs that are available to treat it, and these drugs are not ideal. In most cases, the drugs that are available to treat sleeping sickness are even more toxic, that they kill faster than the disease itself. With the case of Nangana, there are just two classes of drugs that are available to treat it, and the situation here is that of rising resistance to the current available treatment. And Nangana on its own causes a huge economic impact associated with losses in milk production and beef production. And it also makes a vast majority of land, especially in East Africa, not habitable. So you can use it for anything, you can use it for agriculture. And so if you could get a drug that is effective to cure Nangana, you can expand on the agricultural revenue and also get more jobs and the rest. So to this effect, we are interested in developing compounds that can target sleeping sickness, and more particularly in Nagana. Previously, we identified a small molecule abbreviated RB4 with weak activity against the causal agent of trypanosomiasis, which is Trypanosoma brucei. That compound showed a moderate activity of somewhere around 7 micromolar. So we designed new chemistry to generate more analogs to see how we can improve on this activity. And we've appended several heterocyclic compounds and aliphatic units on this particular compound to generate more compounds that we evaluated them for anti-trypanosoma activity. The chemistry of this project was executed by PhD candidate Claudia Angula, and the in vitro biological evaluation is being carried out by the group of Prof. Hope, Henry Hope in Rhodes University. So over 25 compounds were sent there in this series, and we identified that this series was very potent to the causal agent of trypanosomiasis. And the structural activity shows great correlation and established patterns that you can follow to also improve on the activity. For example, we realized in this series that if you modulate the chemistry around position 1 of the quinolone ring, if you increase it with lipophilic units, you get to increase the activity. For example, if you compare compound KT1 and compound KT5, the activity increases as you increase from the ethyl group there to a benzyl ring. And also, if you play around with the lipophilicity, the more hydrophilic, if you put polar groups, you increase activity. Here, the best compound we had here was compound KT7. This compound showed nanomolar activity, wherein you can see against Trypanosoma brucei, the activity there was 80 nanomolar. This compound, however, was not toxic against the human skeletal cells, showing activity almost greater than 10 micromolar, but shows some activity against varus cell line of 3.4 micromolar. Even with this activity against varus cell line, the selectivity is more than 10 units, which makes it an ideal candidate for an infectious disease. The next target that we're interested in is tuberculosis. Tuberculosis is a lung disease that is caused by Mycobacterium tuberculosis. This disease is an ancient disease that has been discovered ever since 1882 by Robert Koch, but presently tuberculosis is still a leading cause of death. Before the coming on of coronavirus, tuberculosis was the leading cause of death from infectious diseases. With coronavirus, COVID-19 now, tuberculosis takes the second place. And it's also one of those risk factors for COVID-19. There are several risk factors associated for tuberculosis, like malnutrition, diabetes, HIV infection, and smoking. Presently, more than 10 million people suffer from tuberculosis each year, and more than 1.5 die from it. And the challenge with tuberculosis is currently the treatment. There are just 4 drugs available for treating tuberculosis. That is the first-line therapy for tuberculosis. And you have to combine all of these drugs into one in the combination therapy, which means if a person is suffering from tuberculosis, he has to take daily a drug load of almost 6 grams, and that has to take place for the next 6 months. And in most cases, after 6 months, people don't get cured because of the emergence of multi-drug resistance, and they have to put them on more therapy for almost over 2 years. And that is really a huge burden on the part of the patients. This burden has, on the other hand, contributed to the emergence of drug-resistant Mycobacterium tuberculosis. And hence there is a need to try to reduce the drug load as well as the duration that people can take the drug. And in order to achieve that, the strategy here is that you have to develop compounds or a compound combination that target more than one physiological state of Mycobacterium tuberculosis, because it is a pathogen that exists in several physiological stages. So to shorten the duration of treatment, you have to target more than one physiological stage. So you have to develop new drug molecules that are polypharmacophoric in their nature. So in order to develop this kind of molecule that can hit multiple physiological stages of Mycobacterium, we are looking at designing new analogs of compounds that have been reported to show polypharmacology. In that, we're interested in nitazoxanide, which bears a nitrofuran. These compounds, they are not that too active. The nitazoxanide, they are not that too active on replicating Mycobacterium tuberculosis, but they have been shown to be active against the dormant stage of Mycobacterium tuberculosis. And if you can hit both the active and the dormant stage of Mycobacterium tuberculosis, you can greatly reduce the duration that is required to get effective therapy, and you can also reduce relapse and hence the emergence of multi-drug resistant Mycobacterium tuberculosis. Another compound class that is of great biological importance is the managed base. Managed bases, they bear both broad biological applications. They are both lipophilic as well as they are both hydrophilic. So they can really enhance good drug-like property for small organic molecules. So in that, we're interested in generating new molecules that incorporate the active pharmacophore from nitazoxanide as well as the managed base. So we want to generate a new compound or new molecule that incorporate this chemistry together. So we've generated or we've designed new synthetic strategies wherein we've generated new series of compounds incorporating all of these pharmacophore that we're interested in. The nitro-thiazole from nitazoxanide that is responsible for the activity against the dormant stage, as well as incorporating the managed base unit. So the synthesis of this series was conducted by Dylan Hart, an MSc candidate. So these compounds were generated, almost 30 of them. We generated 30 compounds and sent them for in vitro biological screening to University of Cape Town. And the person in charge of the biological screening is Prof. Dick B. Warner, University of Cape Town. So they screen these compounds for us. So they start by doing single point. So they screen the compound at a 10-point concentration to generate dose response curves against these organisms. So far we've seen that with this series of compounds, the activity is pretty great. This is a new class of compounds showing some great activity, wherein we have some compounds showing activity from 30 micromolar up to sub-nanomolar, having activity of less than 0.2 micromolar or 200 nanomolar, which is comparable to all the activity of isoniazid, the standard first standard line drug for treatment of tuberculosis. So far, what we've seen is that with this compound class, when we start with a simple molecule like TH11 that has just the nitro-thiazole unit similar to that of nitazoxanide, we have moderate activity of 32 micromolar. But if we increase the activity by incorporating a managed base, as seen from LRO1 up to LRO7, we have diverse activity. And the activity in most cases increases. When you see reduced MIC 90 value, it means the activity of the compound is increasing. So you need just little amount of the compound to get to completely inhibit the growth of the Mycobacterium tuberculosis. So far within this series, what we realize is that if we get substitution at position 2 or 3 of the benzyl ring, we increase activity so greatly. For example, if you look at LRO1, the activity there is 31 micromolar, almost. But when you introduce a methyl group auto to the benzene ring there to give you LRO2, you increase activity more than 10-fold from 31 micromolar to 3 micromolar. And that is also evident when you look at LRO3, where you have substitution at the meter position. So activity goes up to less than 0.2 micromolar. So here we increase the activity almost 20-fold compared to TH11. So this is proof of concept that when we develop a new molecule that incorporates the nitrothiazole moiety as seen in ethazonamide with managed base, when we get a new molecule that incorporates these two pharmacophores, our activity increases greatly. So far, these compounds are promising, and we've evaluated safety profile on ferrosaline, and they showed no activity on ferrosaline, meaning these compounds are exhibiting selective activity against the causative agent Mycobacterium tuberculosis. And these are novel compounds, and being novel means they will definitely be active against the drug-resistant forms as they've not been used before. So just to conclude on the few things that we're doing, we're interested in generating some new molecules against pathogens that are causing infectious diseases. If they are new, they will definitely be active against the drug-resistant forms, as these molecules have not been, as these pathogens have not been exposed to our new molecules before. So far, we've identified compounds against a wide range of disease-causing organisms. Especially with tuberculosis, we've identified several compounds that are as potent as isoniazid, and in most cases, they are more potent than isoniazid, which is a first-line drug for the treatment of tuberculosis. We've also identified new compounds against cryptococcosis. There is a disease that is killing more people and has just 3 drugs in the market. And these drugs are also toxic. And we've identified a new compound class that is active against this disease and is worth further taking forward. We've also identified new compounds for the treatment of Nangana, which are very potent, and they are also worth taking forward. I would just like to thank all of you for listening, and thanks my collaborators in Rhodes University, University of Cape Town, and University of Queensland. And just to mention that the anti-cryptococcal activity, they've been done by the University of Queensland by the non-profit organization, Community for Open Antimicrobial Drug Discovery. And thanks for listening.
Thank you very much, Dr. Betek, for a very interesting presentation. And I have lots of questions for you that I think I might take offline for a bit. We're going to continue directly into the next presenter, and we'll keep all the questions to the end of the session, so we can try and make up some time and get more questions. Get more time for all the questions. So, Dr. Betek, go have some coffee. You'll be up later again for the Q&A. We will then go into the next talk that's going to be by Mututsi Malara, also from the University of Limpopo. And Mututsi is going to be talking about the isolation and structural characterization of oleanolic acid acetate from a plant with a very interesting name that I'm not going to try and say now. Good luck with the presentation.
Thank you. Good day, ladies and gentlemen. I am Marada MP, and I'll be presenting to you my research under the title, Isolation and Chemical Structural Characterization of Oleanolic Acid Acetate from the Leaves of Mystoxylum aethiopica. This study was under the supervision of Prof. P. Masuku and Ms. M. Mwasha. The plant contains secondary metabolites that are known to demonstrate various biological activities and are recently the main focus in drug discovery. This is because plants are readily available and are known to have little or less side effects as compared to synthetic drugs. So the isolation of plant active compound is the first step in developing natural drugs. So the process of isolating and purifying biological active compounds involves the process of bioassay-guided fractionation. So the plant leaves of Misuse aethiopica are used traditionally to treat various ailments such as diarrhea, stomach and respiratory tract infections. The aim of the study was to isolate the biologically active compounds from the leaves of Misuse aethiopica. The objectives were to analyze phytochemicals, antioxidant activity, antibacterial activity, to isolate and purify the active compounds, and lastly to analyze the isolated chemical structure. The plant leaves of Misuse aethiopica were collected at the University of Limpopo. They were then taken to the lab where they were dried and ground to fine powder. The plant material was then extracted using serial exhaustive extraction using solvents of varying polarity, which are hexane, dichloromethane, acetone, and methanol. So these solvents were allowed to extract the plant material for overnight, 3 hours, and for another 3 hours. The phytochemical analysis of the extracts were then analyzed using thin layer chromatogram. So in thin layer chromatography, the TLC plates was loaded with the extracts, which were then developed on 3 mobile phases, which are BEA, CEF, and EMW. The antioxidant activity of the plant extracts was then analyzed using the DPPH radical scavenging assay on the TLC plates. The antibacterial analysis of the plant extracts was then identified against 2 gram negative and 2 gram positive bacteria using the qualitative analysis, which involves the use of the bioautographic assay. So in the bioautographic assay, the developed TLC plate is then sprayed with the microorganisms. The plates are then incubated for 24 hours. After 24 hours of incubation, the chromatograms are sprayed with a tetrazolium salt, which is p-iodonitrazolium violet. So this tetrazolium salt, in the presence of actively growing microorganisms, it is reduced from a clear color into a purple formazan color. So in the quantitative analysis, the cereal broth microdilution assay was used, where we determined the minimum inhibitory concentration of the plant extract. The isolation of antibacterial and antioxidant compounds was carried out using the column chromatography. So 2 columns were prepared in the study. In the first column, the dichloromethane 1, 2, and 3, and acetone extracts resulting from serial exhaustive extraction had high antibacterial activity, so they were combined and subjected to the first column. So in this column, the chemical constituents were eluted using different combinations of hexane, ethyl acetate, and methanol. So the fraction 4 to 9, which consisted of 70% hexane to 10% hexane, and 100% ethyl acetate and 90% ethyl acetate, showed high activity on the first column. So they were then combined and subjected to the second column. So in the second column, the chemical constituents were eluted using 80% chloroform in ethyl acetate. So figure 2 shows the whole summary of the isolation process, where we started with 700 grams of the plant material. It went through the isolation process until we found a pure compound. Table 1 shows the masses of Misuse aethiopica, which were extracted in large-scale extraction, where a total mass of 331 was found, and methanol was found to extracted high plant material of 267, while acetone extracted the least of 7,58 grams. So methanol, because it is a polar solvent, it is known to extract both polar and non-polar plant material. Hence, it extracted more than the other solvents. So the plant extracts, they were then analyzed for phytochemical analysis, where it was found in figure 3A, the compounds that were fluorescing at the wavelength of 365. So most of the fluorescing compound were towards the solvent front. Then the chromatograms were sprayed with vanillin sulfuric acid in order to visualize those compounds that does not fluoresce. So from the chromatogram figure 3B, it shows that the dichloromethane to acetone 3 extracts have similar compounds on the BEA eluent system. So the extracts were then analyzed for antioxidant activity using the DPPH assay. So in the DPPH assay, the presence of antioxidant compounds in the plant extract it is able to reduce the purple color of the DPPH into a yellow formazine color. As it can be seen in this figure 4, the acetone extract exhibited the antioxidant activity. The extracts were then analyzed for antibacterial activity against the 4 microorganisms. So what we observed is that these plant extracts exhibit similar antibacterial activity in all the microorganisms. Hence, I just decided to show one chromatogram. So from this chromatogram, we can see that the dichloromethane and acetone extracts have antibacterial activity, as it can be seen on the BEA mobile phase. So this is seen by the white colored bands. The extracts were then quantified in order to get the minimum inhibitory concentration. So from the minimum inhibitory concentration, it was observed that the dichloromethane extracts 1, 2, and 3, and also the acetone extract 1 had highest minimum inhibitory concentration, which can be seen by the lowest values there. So in MIC, the lower the MIC value, the higher the activity. So these extracts, they were then combined and subjected for isolation on the open column. So from the open column, the extracts were then eluted using different combinations of hexane, ethyl acetate, and methanol. So it was found that the ethyl acetate eluted less plant material as compared to the 50% hexane, which eluted more plant material. So the fractions were then analyzed for phytochemical analysis. So similarly to the large-scale extraction, it was found that the fractions are fluorescing at the wavelength of 365. So there was a greater separation in all the mobile phases. Then when the chromatogram was sprayed with vanillin sulfuric acid, it was seen that 50% hexane to 90% ethyl acetate on the BAL1 system showed to have similar phytochemical compounds. So the fractions also exhibited similar activity in all the tested microorganisms. So I just decided to show one chromatogram. So similarly, on the BAL1 system, we can see that the white compounds there, which shows antibacterial activity, they were active from the 70% hexane to 90% ethyl acetate. So the compounds that were observed on the phytochemical analysis also exhibited antibacterial activity. The fractions were then analyzed for minimum inhibitory concentrations, where it was found that fraction 4 to 9, which consists of 70% hexane to 90% ethyl acetate, showed high activity when tested against all microorganisms. So these fractions, they were then combined and subjected to the second column for further isolation. So a total of 440 fractions were collected from the second column and analyzed for phytochemical analysis. So from figure 8b, we can see that there was a slight difference on the way the compounds were appearing. So we decided to group them into 5 groups of fractions. So those 5 groups, they were then analyzed for phytochemical analysis, where we found that indeed this compound, they show the same compound. So these 5 groups of fractions, they were then analyzed for antibacterial activity. So they also showed a similar activity. So the compounds were then concluded as one compound, and we determined the minimum inhibitory concentration, where the compound showed activity of 250 microgram per mil, as compared to the positive control of 125 microgram per mil. The isolated compound was found to be oleanolic acid acetate. This was concluded based on the information from the NMR and also from literature. So this compound has been isolated from other plants, but except the Mistrosalum aethiopica. In conclusion, this is the first report on the isolation of bioactive compounds from the leaves of Mistrosalum aethiopica. However, this isolated compound has a potential in development of antibacterial drugs due to the widespread activity that was observed throughout the isolation process, but further toxicological studies and other biological activities of the isolated compound are recommended. I would like to acknowledge my supervisors, my collaborators, together with the University of Limpopo and the National Research Foundation. Thank you.
Thank you very much for that presentation and some very interesting results. I think we're going to do the same as with the previous presenter and hold all the questions for the last. So we're going to move on to the next presenter, which is, apologies, let me just get my notes in order again, which is going to be Mashilu Matutoka from the University of Limpopo, who's going to be speaking about the phytochemical content, antioxidant potential, and antibacterial activity of traditional medicinal plants against respiratory tract bacterial pathogens. Enjoy.
Good day, everyone. My name is Matutoka Mashilu. I'm going to be presenting to you a project titled Phytochemical Content, Antioxidant Potential and Antibacterial Activity of Traditional Medicinal Plants Against Respiratory Tract Bacterial Pathogens. I'm trying to skip. Is my screen showing?
Yes, it is showing.
Okay, the sub-Saharan Africa boasts an estimated 45,000 plant species, whereby 45% of these are spread across the Republic of South Africa. It is unfortunate that only 10% of this total flora has been recorded to be used for traditional medicine, although the use of traditional medicine or this form of complementary and alternative medicine has been a long-standing use in South Africa. The success of medicinal plants is based on their possession of phytochemicals. Phytochemicals which have complex structures and hence propel new modes of mechanisms towards dealing with various diseases. It is therefore an interest in the drug discovery to keep a focus or to
Mashilu, if I can quickly stop you, can you just check your microphone? We can't seem to be hearing you. We can't seem to hear you, so if you can just check, please. I think what we're going to recommend is that we move to the next presenter, and then we will try and get your sound back in the background, if that's okay. So my apologies to the next presenter for putting you on the spot, but we're going to move on for now while we sort out Mashilu's sound. So the next presenter is also from the University of Limpopo, and it's Munyadziwa Nelwamondu, and she's going to be talking to us about the antimicrobial properties of medicinal plants used for male sexual health in Limpopo province. Very interesting. Enjoy.
Good afternoon, ladies and gentlemen. I am Ms. M. Arnera Mondo, and under the supervision of Prof. P. Masuku and co-supervision of Prof. M.S. Mposi, I will be presenting to you this afternoon my research project titled Antimicrobial Properties of Medicinal Plants Used for Male Sexual Health in Limpopo Province. Bacterial agents of sexually transmitted disease constitute a significant public health burden in both industrialized and developed countries. Annually, 499 million instances of STIs are reported worldwide, with a further third of these being reported from the sub-Saharan. Pathogens cause many inflammatory disorders within the urogenital tracts, with Chlamydia trachomatis being the most common isolated microorganism. This is one of the most common health issues relating to reproductive and sexual function, as well as one of the leading causes of infertility. Although modern remedies of genital tract infections have been employed, which include ceftriaxone and azithromycin, Waworski and Bemin stated that other patients experience resistance when treated with azithromycin. This could be linked to the overuse of broad-spectrum antibiotics, which can lead to bacterial resistance, inadequate or inappropriate antimicrobial treatment. As traditional medicine develops traction as a viable option to healthcare, screening of medicinal plants for active chemicals is becoming more important. The plants used in the study were selected based on information provided by Mr. Herbert Ramabulana, a herbalist. The first plant is Rhytia natalinasis, traditionally used as an aphrodisiac and treatment of gonorrhea. The second plant is Secudoca longipendiculata, traditionally used for microbial infections and infertility-related problems. The last plant is Elephantiaria burkea, traditionally used as an aphrodisiac and treatment of syphilis. Mr. Ramabulana highlighted that S. longipendiculata, Rhytia natalinasis, E. burkea root material is being used in Zanani, Vembe District, Limpopo Province for sexual incontinence, erectile dysfunction, and sexually transmitted diseases. The aim of the study was to determine the antimicrobial and antioxidant properties of the selected medicinal plants. Hence, the study will only focus on sexually transmitted diseases by investigating the activity of the three plants and the herbal treatment against microbes responsible for some of the infections. Upon collection of the plant material, possible microbial contaminants were identified using the Vitek 2 instrument. These are the identified species, and based on the high identification scores, these identified species are regarded as acceptable. Both gram-positive and gram-negative bacteria were identified as seen in Table 1. This was followed by extraction and phytochemical screening, where in Figure 4, we see the mass of S. longipendiculata, Rhytia natalinasis, E. burkea, and the herbal treatment extracted with solvents of varying polarities. For the three plant extracts, methanol extracted the highest mass, the highest crude mass, and this could be due to the fact that methanol extracts both polar and non-polar compounds. For the herbal treatment, water extracted the highest crude mass, and this further validates the ingestion of the herbal treatment using water as prescribed by the herbalist. Qualitative phytochemical was determined using standard chemical test and thin layer chromatography. In Table 2, we see the phytochemicals which were screened for, and all these phytochemicals were found to be present in the herbal treatment and in all the plants. This was followed by the phytochemical profile of each plant, which was analyzed using the back thin layer chromatography, and they were developed in different eluents, namely BEA, which is nonpolar, CEF, which is intermediate polar, EMW, which is polar. And then they were visualized under UV light of 254 nanometers in A, in 365 nanometers in B, with an interest to fluorescing phytochemicals, which were visualized in the BEA system of the Elephantiaria burkea plant extracts. They were further sprayed with vanillin sulfuric reagents, where once more the BEA eluent system showed phytochemical phytochemicals, which are seen with the pigments of purple and red colors. Quantification of the major phytochemicals was performed where the total polyphenol content was quantified colorimetrically. For the flavonoid content, the herbal treatment showed the highest content through the hexane extract with a value of 904 quercetin equivalents per gram of extract, followed by Elephantiasis burkea, once more with the hexane extract with a value of 204 quercetin equivalents per gram of extract. For the phenol content, Escherichia pendiculata showed the highest content with the hexane extract with a value of 159 gallic acid equivalents. And for the tannin content, the Escherichia pendiculata content once more showed a high content with the hexane extract with a value of 157 gallic acid equivalents per gram of extract. Qualitative antioxidant assay was performed where the presence of antioxidant activity was indicated by the development of yellow bands on TLC plates against a purple background of the DPPH, which was reduced to a yellow color, which is the formazine. So in figure C, we see the chromatogram of the medicinal plants in the herbal treatment extracted with solvents of varying polarities developed in BEA, CEF, EMW mobile system, and then sprayed with 0.2% DPPH in methanol. Elephantiasis burkea showed antioxidant activity in the EMW Lewin system, but the more resolved compounds were seen in the CEF mobile system. Antioxidant activity was further quantified using DPPH. And in figure 7, we see the percentage free radical DPPH inhibition of Escherichia pendiculata and W. natalinasis at varying concentrations measured at absorbance of 570 nanometer. Ascorbic acid was used as the standard. For Escherichia pendiculata, hexane extract showed the highest percent inhibition. For W. natalinasis, methanol extract showed the highest percent inhibition. In figure 8, we see the percentage free radical DPPH inhibition of E. burkea and the herbal treatment at varying concentrations measured at absorbance of 570 nanometers, and ascorbic acid was used as the standard. For Elephantiasis burkea, the hexane extract and the acetone extract showed the highest percent inhibition, and they reached a maximum threshold at roughly 20 micrograms per milliliter. And the dichloromethane also showed promising percent inhibition as it was competing with the ascorbic acid. For the herbal treatment, the dichloromethane showed the highest percentage inhibition. This was further checked for the ferric reducing power of the Escherichia pendiculata and the Wrightian natalinasis at varying concentrations expressed as absorbance of 700 nanometers, and ascorbic acid was used as a standard. For the Escherichia pendiculata, hexane showed the highest absorbance, and for the Wrightian natalinasis, hexane once more showed the highest absorbance. In figure 10, we see the ferric reducing power of Elephantiasis burkea and the herbal treatment at varying concentrations expressed as absorbance of 700 nanometers, and ascorbic acid was used as the standard. The hexane extract and the acetone extract showed high reducing powers, and the hexane reached a maximum threshold at roughly 55 micrograms per milliliter, whilst the acetone reached a maximum threshold at roughly around 125 micrograms per milliliter. And once more, the dichloromethane was competing with the ascorbic acid. For the herbal treatment, the dichloromethane showed the highest absorbance. This was followed by antimicrobial activity, And the test organisms which were used were gram-positive Enterococcus faecalis, Staphylococcus aureus, and for the fungus, Candida albicans was used because it's associated with STI, which is called candidiasis. So bioarthrographic investigations were done for qualitative antimicrobial assays, and for quantitative antimicrobial assays, minimum inhibitory concentration was done, and the antibiotics used were gentamicin and amphotericin B. Further synergistic and antagonistic studies were analyzed using the fraction inhibitory concentration. In figure 10, we see the chromatogram of the medicinal plants and the herbal treatment extracted with different solvents and developed in BEA, CEF, and EMW mobile systems. And they were sprayed with E. faecalis in A, they were sprayed with S. aureus in B, and sprayed with C. albicans in C. The bioartographic investigation on the TLC plays was carried out to discover the major bioactive chemicals within the crudes, where the compounds of the antibacterial or antifungal activity is shown with the inhibition of the clear zones. As seen in A, they were found to be in BEA and CEF, and in B, for the S. aureus, they were found in BEA, and in C, for the C. albicans, they were found in CEF. So the E. faecalis had similar profiles in both the S. aureus and the C. albicans. This was followed by the minimum inhibitory concentration of the antimicrobial of the plants, where the plants were observed for clear wells, which is regarded as activity, which resulted from the reduction of the purple color of the tetraxylum salt, and the MIC values were recorded as the lowest concentration that the bacterial growth inhibits. That was noted on the Elephanteriza burkia plant for the hexane extract and for the acetone extract. And the MIC values of the fungus was noted on the Elephanteriza burkia for the methanol and for the water extracts. The total activity of the medicinal plants and the herbal treatment was further analyzed against the bacteria, and Escherichia pendiculata showed high activity on the methanol extract for both S. aureus and E. faecalis. And for the fungus, the C. albicans, the high activity was noted on the Elephanteriza burkia plant on the methanol extract. Combinational effects of the addition of combination species on antimicrobial activity was investigated, and the outcomes were either synergistic, additive, indifferent, or antagonistic. And in table 9, we see the antibacterial activity of extract combinations against Staphylococcus aureus, where most of the outcomes were indifferent or antagonistic. However, 2 of these combinations were found to be additive, which contained the Wrightia natalinacis and the Escherichia pendiculata, the Wrightia natalinacis and the Elephanteriza burkia. In table 10, we see antibacterial activity of extract combinations against Enterococcus faecalis, where most of the outcomes were found to be antagonistic and indifferent. However, Wrightia natalinacis combined with Elephanteriza burkia showed an additive effect. In table 11, we see the antifungal activity of the extract combinations against the Candida albicans, and all these outcomes were found to either be antagonistic and indifferent. Elephanteriza burkia exhibited high antioxidant activity and noteworthy antimicrobial activity, and based on the good correlations found in Ehrlichia, it was selected for bioactivity guided isolation of antibacterial compounds. And the study suggests that the evaluated plants are potential sources of anti-infective agents. Future works to still be done is the isolation of the bioactive compounds and cytotoxicity tests. I would like to acknowledge the following people and the following bodies. Thank you.
Thank you very much for that presentation. I must say I learned a lot, which was exciting. So expect an email from me if we don't have enough time for questions, please. We're going to quickly go back to the previous presenter and see if Mashilo can continue. He had some internet issues, so let's try and give that a second go. Good luck, Mashilo. Mashilo, we still can't hear you.
Can you hear me now?
Yes, we can hear you now.
Am I audible?
Yes, we can hear you.
Am I audible now? Yes, I can hear you. Can you hear? Hello. Hello, am I audible? Hello, everyone. Hello.
Okay, perfect. While they sort out Mashilo, let's go on to Wanda Shekwa. Wanda's going to be talking about the antimicrobial and antioxidant activity of Cariso bispinosa leaf. So good luck with this, and sorry for throwing you into an early position as well.
Good afternoon, everyone. My name is Wanda Shekwa, and I'm going to present to you the part of my research which is titled Isolation and Identification of Antimicrobial Compounds from the Leaf Extracts of Cariso bispinosa used for dental health care. The research was conducted under the supervision of Professor Masuku from the University of Limpopo. Dental diseases are amongst the most prevalent diseases worldwide. About 49 to 83% of the people worldwide are affected by dental caries. People who are affected by dental diseases may have a low self-esteem and may have discomfort when speaking, eating, and drinking. The current dental treatment is facing several disadvantages, including toxicity, low efficacy rate, limited number and high cost of medication, serious side effects, and antibiotic resistance. Cariso bispinosa is one of the medicinal plants used in South Africa in the treatment of toothache. The aim of the study is to isolate and identify the antimicrobial compounds from Cariso bispinosa's leaves extract, with the objectives being to extract phytochemicals, determine phytochemical profile, to determine antioxidant and antimicrobial activity, to determine cytotoxic effects, to isolate and identify the antimicrobial compounds. A series of methodologies were performed to obtain the objectives, where extraction was achieved using solvents of varying varieties, which are hexane, chloroform, dichloromethane, ethyl acetate, acetone, ethanol, methanol, and water. And the phytochemical profile was generated using the thin layer chromatography. The antioxidant activity was screened using TLC-DPPH assay, and it was quantified using the DPPH free radical scavenging activity assay and the ferric reducing power assay. The antimicrobial activity was determined using bioautography and minimum inhibitory concentration assay. The cytotoxicity effect were determined using MTT assay. The isolation of the antimicrobial compound was achieved using the open column chromatography and preparative TLC. And the identification of the compound was done using the nuclear magnetic resonance. The following are the results which were obtained after the series of experiments were performed. The graph in figure 1 shows the mass extracted from 1 gram of leaf powder using solvents of varying varieties. And from the graph, you can see that the extract that the solvent that extracted the highest mass was methanol, which is a polar solvent, and that is accounted for by the ability of polar solvents to extract both the non-polar and polar phytochemicals. And figure 2 shows the phytochemical profile of the leaf extract of Calisapis spinosa. The chromatograms were developed in the BEA, CEF, and EMW mobile phases and analyzed under UV at 264 nanometers and 365 nanometers, and further sprayed with vanillin sulfuric acid to visualize the non-fluorescent compounds. And from the figure, we can see that the bands are visualized in all the mobile phases. However, better visualization was observed after spraying with vanillin sulfuric acid, and the mobile system that separated the bands better was the BEA mobile system, which is a non-polar solvent system, suggesting that most of the phytochemicals may be non-polar. The antioxidant activity was determined using the TLC-DPPH assay, and the yellow spots in the purple background represent antioxidant activity. And from the figure, we can see that the spots are present in all the mobile phases. However, a little separation is observed only in the EMW mobile phase, suggesting that most of the antioxidant compounds may be polar. The antioxidant activity was quantified using the free radical scavenging activity assay. And from the graph, you can see that most of the polar extracts are the ones which are having the highest antioxidant activity. As you can see, the methanol is quite high, followed by the ethanol. The ferric reducing power assay was also used to confirm the results which we obtained previously on the DPPH free radical scavenging power assay. And we can see also in this graph that the polar solvents also exhibit the highest antioxidant activity. The antimicrobial activity was determined using the bioautography assay, where the chromatograms were also developed in the BEA, CEF, and EMW mobile phases and tested on S. aureus, S. pathogens, and E. faecalis. As we can see from the figure that there is a white band in the pink background which represent antimicrobial activity, and the activity is constant, is similar in all the chromatograms, which means there's a single compound that is active against all the pathogens. The antimicrobial activity was quantified using the minimum inhibitory concentration assay. And from the table, we can see that the most susceptible microorganism was Candida albicans, which was inhibited on average by 0.73 milligram per mil. However, the most effective extract was the methanol extract, which could inhibit the microorganisms at on average at 0.81 milligram per mil. The cytotoxic effects were determined using the MTT assay, and from the graph we can see that the percentage viability was concentration dependent, where an increase in concentration leads to a decrease in cell viability. However, at a concentration of 0.25 milligram per mil, which is a same concentration that led to antimicrobial activity, the extract was still not toxic, with the percentage cell viability being still high, which suggests that the extract may be safe to use for human consumption. Open column chromatography and preparative TLC were performed to isolate the compounds, and this isolation was guided by the bioassays. And after isolating the compound, it was found to be a fine powder, and it was sent to the Department of Chemistry in the University of Limpopo for NMR analysis. The spectra that was generated was sent to Prof. Mazimba in Botswana, who analyzed the compound and generated the structure and elucidated the structure of the compound. Figure 9 shows the antimicrobial activity of the isolated compound. The chromatograms were developed in PEA and also tested on E. faecalis and S. aureus for antimicrobial activity. And the activity was also observed with a similar patterns from the previous chromatograms. The antimicrobial activity was also quantified using the MIC assay. And from the table, we can see that the compound was effective against both E. faecalis and S. aureus, where E. faecalis was inhibited by 250 microgram per mil, and S. aureus was inhibited by 125 microgram per mil. Figure 10 shows the structure of the isolated compound, which is known as beta-sitosterol, a member of the plant phytosterols. In conclusion, the study has demonstrated the presence of antimicrobial compounds from the leaf extract of Carisalis spinosa, and it is the first to report on the isolation of beta-sitosterol from the leaves of Carisalis spinosa. However, further studies should be done for a possible development of drugs from the compound. I'd like to acknowledge the following people: Dr. Mazimba, Ms. Selani Ramakari, my colleagues at the Department of PMBT, and the University of Limpopo, along with the CSIR. Thank you.
Thank you very much, Fonda, for that nice presentation. We are going to try and get Mashilo back, so don't run away. We may still get you for some Q&A, but we're going to try Mashilo one more time before we go to Q&A. For the participants, stay with us, please. We know we're getting closer to lunch. maybe get Uber Eats to start delivering or order people around your house to get your lunch made. But we are going to try and leave you with a decent amount of time for lunch as well. Good luck, Mashilo.
Hello, everyone. Am I audible? Everyone. Hello, everyone. Am I audible? Okay, thank you. Just the fact that there are a lot of plants which are used traditionally and they have not really been recorded yet as being used. So therefore, that is why drug is of interest, with a mission to try and obtain safe, efficacious, and affordable therapies for these increasing human diseases. It was therefore the aim of the study to determine the antibacterial, antioxidant, and anti-inflammatory activities of this medicinal plants used for the treatment of respiratory tract infections. The selection of these plants was based on the ethnopharmacological use. That is to say, that were based on how various cultures use medicinal plants towards treating various ailments, particularly for the study.
Went a bit chaotic, so we're going to try and see if we can't accommodate Mashilo at another stage in the sessions. We'll try and make sure we see if we can't help him that way. We still have a few seconds left, so let's see if we have any questions from the floor for the presenters that just did their presentations. Do we have anybody? Questions so far. I'm just checking the question and answer box. So there's nobody at this stage that have questions. Is everybody already on their way to the kitchen? Oh, there we have Dr. Eunice.
Hi, Tobias. I just wanted to find out from Modiswa, the lady who did the work in Limpopo, how she went about selecting the organisms that she tested against, because I think most of them were gram-positive, but obviously some of the diseases she was talking about are from gram-negative organisms. So how did she select that, and would she be possibly doing some further work with some gram-negative species as well?
Okay. Was that for? Sorry. Eunice? Oh, there you go.
Hi.
Can you answer for us? Thanks.
Thank you for the question, Dr. Eunice. To answer you, the selected microorganisms was the C. albicans, which is a fungus associated with the STI known as candidiasis. And then you are correct, the bacteria which were chosen were gram-positive, and they were also found in semen, in isolated. They were isolated rather in semen of males with STIs as well. But yes, gram-positive, gram-negative rather, bacteria will also be studied.
Okay, thank you so much, Munyadiswa. I Hope I got your name right.
Thank you.
I actually had a similar question to Wanda, where the organisms, I'm not a dental specialist, but I was wondering, Wanda, the organisms you tested, are they associated with infections of the mouth, or would you at some stage look for similar for organisms that would cause infections in the mouth?
Okay, thank you for your question. The organisms which were used were Candida albicans, which is responsible for oral candidiasis, and Candida glabrata was also used, which is also a cause of oral thrush. So the other organisms which we use are transient pathogens in the mouth.
Okay, so but will you look at more common pathogens in the future?
Yeah, the most common is Streptococcus mutans, and we are going to check it as well.
Okay, cool. Thanks.
Okay.
Do we have any other questions for the presenters? Nothing, it seems. With that, I think we're going to end the session. It's 1 o'clock on my computer now. You've got 30 minutes for lunch. Professor Vasal Khan will take on the next session at 1:30. So please try and log in about 5 minutes before the time, get ready, get settled, so we can continue. Thank you to the presenters for the session. It was really interesting talks. And look out for some emails from my side with some questions for help. Thank you, everybody.
Okay, thank you very much. Good afternoon, everyone, and welcome to the final session for day one, where we are going to be focusing on environmental health. Before I welcome our keynote speaker, I would just like to go through a few housekeeping rules. I would appreciate it if all the presenters could please try to adhere to the time limit of 15 minutes. We have 20 minutes for our keynote speaker. Please also keep your microphone and video off until it is your turn to present. For the audience, you are welcome to type your questions in the chat room, and we will address all the questions during the question and answer session. So it now brings me great pleasure to introduce Dr. Jo Barnes as our keynote speaker for this session. Dr. Barnes is a senior lecturer emeritus in epidemiology and community health of the Faculty of Medicine and Health Sciences at Stellenbosch University. She is a member of the Berg Catchment Management Forum and the Pollution Task Team of the Berg River Irrigation Board, as well as a consultant to the task team to determine additional resources needed to manage pollution in stormwater and river systems of the city of Cape Town. She is the recipient of multiple awards, amongst them the Order of the Dizer, a member class in 2007 for meritorious services to the province of the Western Cape, winner of the Women in Water Sanitation and Forestry Award in 2007, winner of the Cape Times Caltex Environmental Award in 2005 for the research work on contaminants in rivers, and recipient of the Faculty of Health Sciences Award for Community Service in 2007. Now, she and her students have studied community health in dense and low-cost settlements for many years, and as a prominent epidemiologist and community health specialist, she has and continues to publish numerous peer-reviewed articles, popular publications, and she has presented multiple keynotes and plenary presentations. Her CV is obviously truly impressive. And Dr. Barnes, thank you so much for agreeing to present a keynote address, and we look forward to your presentation. Over to you.
Good afternoon, everybody. This has been a highlight of my career, and I'm so happy to be here because I get to say thank you today to all the magnificent work that I've been hearing this morning and that will be presented in the next few days. I suspect I've been asked to talk to you. I have no visuals, it's just me. I was suspected that it was a sort of an Auntie Mavis presentation, you know, a wisdom from Auntie Mavis. So it is supposed to be called AMR in the water sector. We have come some way, but we have far to go. But I am not at all surprised if you think that this is just a lesson from somebody who walked a long way. I have 3 pieces to my presentation. One is some history to show you how far we have come professionally and how proud we should be of that. The second part is some of the urgent needs that are neglected and had been neglected for so long. Some of them you are picking up already today and tomorrow, just as an inspiration for the new generation so that I can hand over and go and do some knitting. And then there's some wise advice for scientists in managing your career and managing yourself, perhaps more aimed at the younger ones, but it's always good to be reminded of some stuff in the heat of the moment. So let's have a look at the case history of the early career of Jo Barnes. I graduated with a degree in mathematics and couldn't find a job at all. So eventually I found a job as a filing clerk at the branch of the Department of Agriculture. While I was filing letters and reports, I applied for posts at, let's tactfully call it, government-connected institutions with this degree in mathematics. So dutifully, they let me know that I have to present myself at a certain place at a certain time so that I can write an IQ test. Okay. Whatever it takes. So I went along to this hall to write the IQ test, and nobody said anything, but there were only women in the hall who had to write those tests. And about 3 or so weeks later, I got a phone call from both these institutions to say, unfortunately, they cannot offer me a post because I did too well in the IQ test. Now, it's the first time I've ever heard that you can fail a test upwards, but I did, I managed. The one said that the head of the division where I was supposed, where the post is, felt that I would be bored. So I said, do you think filing letters is exciting? But no go. I deducted that I probably had a higher IQ than him, whatever. Here we sit today. We have none of that anymore, hopefully. No, nobody makes women write IQ tests before they can get a job. I am so grateful for where we are, and we should take a moment to say thanks for that. We have other issues. The not being able to get a job is very familiar to many people, and I'm really concerned that scientists in South Africa working in academia often, particularly the early career scientists, have no job security. We have too many people in South Africa with degrees driving Ubers, and that has to change. We have to make space and opportunities for the people that we train and graduate. They have a huge lot to offer us and the country. We cannot treat them like that. So that was point number one. I wanted to say that some of that, and please help me because you've got bigger voices than I have. I'm just Auntie Mavis by now. I went back to filing, but I picked up there plenty of times in the day that I had nothing to do because I filed too fast, you see. Picked up on documents on water and water pollution, and I was starting to be fascinated, and I read them. Never think if you parked in a corner and life took a sideways with you that opportunities are not there. Opportunities hide in unexpected places. I read those things. I learned an enormous lot about water pollution, and it sparked a lifelong interest for me. Opportunities and sideways moves and temporary setbacks are not the end of the world. Believe in yourself and learn whatever skills you can add. I've never learned whatever humble skills even that I didn't afterwards find a place to use. So the end of the first part of this is to say to you, I wish I could tell you that life gets better. Life doesn't always get better. You get better. So let us go on to the second piece, that is the inspiration for the new generation. By the way, I am so proud of what I had been seeing so far and what is going to be presented. This is a gold mine of information and skills and approaches that I really would like to urge you to not only publish in a journal overseas, but to talk about it wherever you can, present it in some more simple form to larger sections of the population. I'm not asking to turn you into an activist, just bring your work to the outside attention. It does your career no end of good, and it helps you to gain traction so that you can become more easily employable for that, and it will expand the profession for not only for you but for your co-workers as well. Those early documents that I read at agriculture inspired for me a lifelong interest in the environment and in water. But as an epidemiologist, I was fascinated by the opposing roles of water. Beyond sustaining life, it's one of the best sanitation devices that we know. It is a major agent for cleanliness, but on the other side, it's a major carrier of disease and environmental degradation. Water does what we treat it like. And we are not good at that at all. We have 824 wastewater treatment works in the country. Fewer than 10% of them are delivering water of a safe quality safe for humans and safe for the environment. That is a shocking statistic. I'm sorry, but that is a human rights transgression. At least 1 in 4 municipalities are dysfunctional in the country, and 2/3 of them are in financial distress. Partly the reason why we have this massive failure of wastewater treatment works. And one of the nearby major towns, I was working in the dense settlement at that stage, and I was pleading with a member for the town council for that area to please vote, help me so that we can vote more money to improve the toilet facilities and the sanitation and the sewerage system in the township. He looked me straight in the eye and said, there are no votes for me in sewage. We need to change that. We really need to change that. Coupled with climate change, we are facing a huge number of inappropriate discharges and also high risk of reuse of poorly treated water, grey water included. May the saints preserve them, but many of these municipalities who said chuck the grey water on the vegetables in your garden got no idea how small they make that infection cycle from the infected people in the house onto the vegetables and back into the house. It's an incredibly small and fast cycle. I'm really, really concerned about the logical but flawed argument about reuse of water will solve all our problems if they don't even know what's in that water after they have so-called cleaned it. I'm happy to see that there's some of that as well that makes me sleep a little bit easier at night as well. We did in 2002, long time ago, we did some small chlorine resistance tests for E. coli. Those were E. coli we just isolated from the Eerste River system, and much of it comes from completely untreated sewage. So they were natural organisms. And I want to credit my lab technologist at the time and say, Joe, let's have a look at the AMRs of the E. coli that we pull through the chlorine and see what happens to that. And my goodness, the ones who survived the chlorine experiments just about doubled their resistance to we tested them before and after, just about doubled their resistance. So municipalities who improperly chlorinate the effluent at the treatment works are creating not just one problem, but two. They are not only releasing poorly treated or unscathed organisms into the environment, but they are now very much more resistant as well. And I never had the opportunity or the money to actually follow up on that. I'm seeing some of that work and I am really so happy to see that. I just want to encourage you to do more of this kind of work, to look at the consequences of poor execution of duties. I also have serious concerns about the inappropriate use of antimicrobials as preservatives in foodstuffs. I picked up a punnet of raw mincemeat in a supermarket a couple of days ago, and my goodness, there are mycins in there as a preservative. And it's not directly in your remit, but a lot of these inappropriate uses of antimicrobials are for preservatives and as production enhancers in animal production, etc. Is now not only causing AMRs, but it's also causing an upsurge in serious and sometimes life-threatening allergies in some people. So now we're again ending up with doing something, and it's not only one intended consequence, but two unintended consequences from it. So there's also some recent concerns have been emerging of the use of the new antiviral medications for treatment of COVID-19, particularly the one from Merck. The fear is that it may actively encourage the virus to evolve more variants, but that it also may end up in some kind of AMR situation. It's not yet, it's unethical at the moment to withhold those treatments from gravely ill people on the basis that it may cause antimicrobial activity. But it is something that I worry about, that we are in the situation. And I want to support a shift in the blame game, as I call it, for instance, that AMR, it's mostly in the rich countries, affluent countries, mainly in the north, that AMR is being mostly caused by wrong use or overuse of antimicrobial medication. We down here in an area where there's a lot less resources also end up with a scenario where poor functioning of our various systems are causing AMRs in places and in people who've never seen an antibiotic in their lives. So we don't know what acid mine drainage, microplastics, chemicals of emerging concern, overuse of certain preservatives, disinfectants, sanitizers, triclosan, anybody? Are all contributing to AMR, while our regulatory capabilities to control some of that stuff, it doesn't help to have rules if you don't apply them and oversee them. So just before I finish off, I have a few bits of wise advice, especially for our younger presenters, and I am so immensely proud of those that I have seen today. Do not lose heart when life takes sudden turns, side turns. How you think when you lose determines how long it will be until you win again. And don't ever forget that you are special and you have special skills. You have a unique collection of skills because life walked a route with you up to now. Just make sure that you add to those skills every which way you can. There is another saying that I got on my fridge to keep my feet on the ground. Talent is God-given. Be humble. Fame is man-given. Be grateful if it happens to you. But conceit or arrogance is self-given. Be careful. My last little bit that I want to add to this is the tennis player Arthur Ashe he acquired HIV from a blood transfusion before anyone knew that it was possible that it could happen. He spent the rest of his then shortened life trying to do whatever he could for his fellow mankind in his community. And he had a saying, because you're not going to get all the resources you need. You're not going to be able to do what you wish. By the way, sometimes it's better not to get what you wish, because imagine if I still had to do some calculations in a back room for somebody, I would have died. He had a saying, start where you stand. Do what you can, use what you have. Just keep at it, and the opportunities will come by. I want to say thank you to you because you gave me 20 minutes of your life today. I hope it was a good investment. And my congratulations on the work reported here at this symposium. I'm again so proud of you. And what is happening is that you make me sleep more easily because I worry about these things. I'm deeply grateful to Professor Besaidnout from NWU and Dr. Kalabaila from the WRC. And congratulations for the WRC on their 50th anniversary, for this invitation, and for the organizing committee and for all involved. You would be easily able to find my email address. If you want to send me a mail and you want to ask something, I'm not all wise. I made massive mistakes all the way, which is why I can sit here and sound so smart. But if I can support you, if I can help you, let me know, and I promise I will answer. And thank you very much for all of this, and my very, very best wishes to you. Thanks.
Research experience. You are truly an inspiration to all of us. And while we are going to hold over the questions to the Q&A session, I really want to encourage the younger scientists to use this opportunity to pose some career advice questions to Dr. Barnes. Thank you again, Joe. Okay, that takes us to our next speaker, who is Ruan Pori from Northwest University. And Ruan is going to be presenting on the antibiotic-resistant Clostridium perfringens in rivers.
Okay, hello everyone. My name is Ruan Pori. I am a student at Northwest University, and I will be talking about antibiotic-resistant Clostridium perfringens in river systems, a phenotypic and genotypic approach. So just a brief overview of Clostridium perfringens. It is an anaerobic gram-positive endospore-forming bacteria that can be found in various environments ranging from clinical to environmental settings, as well as the gastrointestinal tract of humans and animals, and sewage effluent of wastewater treatment plants. Now, Clostridium perfringens is one of the more well-known pathogens in this genus and cause a variety of illnesses such as gas gangrene and food poisoning. Now, the treatment for these infections are a prescription of antibiotics, the most commonly being beta-lactams, clindamycin, or metronidazole. But with the ongoing worldwide problem of antibiotic resistance, it's becoming less effective, antibiotics are becoming less effective for treating these infections. And this is mainly due to the overuse and misuse of antibiotics. Now, the contributing sectors to this ever-growing problem is the medical field for both human and veterinary medicine, aquaculture, livestock, and agriculture, where they use antibiotics as growth promoters. Now, anthropogenic factors that contribute to antibiotic resistance, especially in the aquatic environment, are agricultural runoff and industrial runoff, as well as poorly treated sewage effluent that is introduced into this river system. Now, very interesting to note is that antibiotic levels in surface water are less detectable than that in aquatic sediment because of the antibiotic residue is more stable in the sediment, resulting in them staying active for longer periods. Now, when we were doing our due diligence, we saw that there has been studies done in Clostridium perfringens in the water and sediment section, but its antibiotic resistant traits have only been partially explained. Now, this is mainly due to inadequate laboratory conditions or uncertainty about the current methods used to cultivate and enumerate antibiotic susceptibility and anaerobes, specifically Clostridium perfringens. Now, a possible solution to this is actually the Fung double tube method. Now, this method was used for quality control in food stuffs, where they cultivate and enumerate obligated anaerobes such as Clostridium. Now, this method achieves anaerobiosis by no additional atmospheric generators or chambers, where you add your liquefied media with 1 ml of water sample in this case, and then you insert an insert tube into the larger test tube, but then it creates a thin layer between the test tubes with no or minimum headspace. Now, we adapted this method for isolating antibiotic-resistant Clostridium perfringens from aquatic environments by supplementing the media with antibiotics at the CLSI breakpoint concentration. And it is more convenient, cost-effective, and time-efficient than your traditional method. So the aim of the study was to isolate antibiotic-resistant Clostridium species from various various river systems in the Northwest province by using this modified nwu-pukke method, and determine the phenotypic resistant profiles and utilizing whole genome sequencing to investigate antibiotic resistant traits. So the two river systems we investigated is the Crocodile River and the Squinspate River in the Northwest province. These rivers in total had, between the two rivers, we had a total of 13 sampling sites. Now, as you can see through the sampling images here, these river systems are not in pristine condition. There's a lot of anthropogenic runoff factors like agricultural runoff from neighboring farms, as well as a lot of littering. And you can see the end of bottom right, the Squinspate River of the organic foam indicating very nutrient-rich environment. And this is just outside of the wastewater treatment plant. So a brief overview of the materials method. So we isolated the Clostridium differentians from the sediment and surface water using the nwu-pukke method, supplementing it with ampicillin, tetracycline, and clindamycin. We identified them through 16S RNA gene amplification, and then we did agar dilution to determine the resistant profiles. A couple of isolates were chosen to do whole genome sequencing on. We did the assembly and annotation, and we used a couple of pipelines to determine the presence of antibiotic-resistant genes. So this is an example of the results that we achieved from the river system. This is surface water. You can see the black colonies now are antibiotic-resistant Clostridium differentians species. I think this was against ampicillin, and here tube 5 was the effluent that's being introduced into the river system, and you can see there's definitely an overgrowth of antibiotic-resistant Clostridium around the system there. So among the 67 Clostridium differentians isolates, 68 were resistant to ampicillin, followed by tetracycline and clindamycin. Clindamycin had also the highest MRC concentration, with 33% of all isolates being able to grow at concentration above 256 micrograms per milliliter. Now, metronidazole showed the lowest MRC value, just below 50%, where more than 72% of the isolates were highly susceptible to amoxicillin and chloramphenicol. Now, although most of the isolates demonstrated resistance against ampicillin, they also showed most susceptibility to amoxicillin. So this could indicate the variation in the beta-lactam resistant phenotypes among these isolates. So out of all the, oh, sorry, let me just go back here, thank you. Out of all the antibiotic-resistant Clostridium differentians isolates, 28 of them were resistant to 3 or more antibiotics. All the multiple resistant isolates were from either downstream from the wastewater treatment plant in the Squinspate River, except for one isolate that was obtained from the Crocodile River. The only antibiotic that was constantly present in all the MAR phenotypes was ampicillin, as shown here in table 2. The most dominant AMR phenotype was ampicillin, tetracycline, clindamycin, metronidazole, chloramphenicol, and amoxicillin. So a total of 9 isolates presented this phenotype. All these isolates were obtained from sediment at the downstream from the wastewater treatment plant. This highlights that sediment in rivers which are exposed to antibiotic pollution, probably from the effluent of the wastewater treatment plant, can harbor large quantities of multi-antibiotic resistant bacteria. So from there on, 3 isolates were chosen from surface water to do whole genome sequencing on, and we used various pipelines and bioinformatics tools to determine the potential of the presence of antibiotic resistant genes in there. So using tools such as deep ARG, we could identify a total of 11 antibiotic resistant genes in the genomes of the 3 Clostridium differentians isolates. isolate 13 and 17 both possess genes that encode for resistance against 7 different classes of antibiotics. This included beta-lactamase, macrolide lincosamide, streptogramin B, tetracycline, trimethoprim, I think there's one more. Whereas 24 only harbored resistance against 6 classes with the absence of the beta-lactamase resistance. Also important to highlight that all 3 of the isolates had 2 or more genes encoding for multi-drug resistance. So the average nucleotide identity of the Clostridium perfringens genomes were successfully calculated using AutoANI. The analysis of the 3 isolates showed that showed them to be almost exactly identical, but when compared to the 3 Clostridium genomes, so when comparing the 3 Clostridium genomes to that of the 3 reference genomes, another high similarity was observed among genomes, with our genome sharing an ANI value of 98.5 to 98.6% with the reference genomes. This dendrogram also shows that the 3 isolates were clustered more closely to the 025 and the 80cc strain, which was derived from clinical environments, as to the strain 13 was originally isolated from natural environment, specifically soil. So some of the conclusions we could have made here is that the single tube method was actually very successful in isolating antibiotic-resistant Clostridium perfringens from these environments with the supplementation of various antibiotics at its clinical breaking point. Now, in these environments, the most resistance was shown to tetracycline and ampicillin, which is one of the most prescribed antibiotics, and the most susceptible was amoxicillin and chlorphenical. There were some isolates that were showed resistant to all of these antibiotics, which is really worrisome. And also very important is that sediment is a reservoir for antibiotic-resistant Clostridium perfringens in these aquatic environments. And the presence of ARGs encoding for several classes of antibiotics and multi-drug resistant efflux pumps could contribute to the emergence of pathogenic multi-drug resistant Clostridium perfringens in this environment, as well as the comparative genomics showed great similarities between the environmental Clostridium perfringens strains with those of clinical origin, therefore highlighting the importance of monitoring these bacteria for effective epidemiological surveillance purposes. It's also very necessary to screen for antibiotic resistance in these aquatic environments to further investigate potential sources that can arise within them. And all of this is very concerning because what we found throughout the study when we were sampling is these river systems serve rural and informal communities where they directly depend on these water sources. And especially for immunocompromised individuals and even livestock that use this water, they might be at risk. And also these river systems are used for irrigation for the neighboring farms, and this can contribute to the spread of antibiotic resistance, specifically Clostridium perfringens as well, through other niches. So yeah, I want to thank you for listening, and I'd like to thank the support of the NRF, Water Research Commission, and ARC for funding this program.
Okay, everyone, thank you very much. Very interesting research. I have a few questions, but I will wait for the question and answer session. Thanks, Rowan.
Thank you.
Our next speaker is Tshepiso Masiola, also from Noordwes-Universiteit, who is going to be focusing on antibiotic resistance profiles of Enterobacteriaceae isolated from groundwater intended for human consumption. Thank you very much.
Good afternoon, everybody. My name is Tshepiso Masiola. Can you please give me a minute? Good afternoon, everybody. My name is Tepiso Masola, and I will be presenting to you the topic of prevalence of quinolone-resistant profiles among Enterobacteria zeae isolated from groundwater that was intended for human consumption. This was a project that was done in 2019 in fulfillment of an honors degree. Globally, groundwater represents the world's largest and most important source of fresh potable water. In South Africa, groundwater is highly used in rural and arid areas. Mafikeng area, being one of the most arid areas in the country, faces challenges with water shortages as well as poor community service. This results in most community members of Mafikeng being reliant totally on groundwater for fresh water, as a source of potable water. But groundwater is unprotected and is easily contaminated through sewage runoffs, urban runoffs, as well as agricultural runoff. This introduces microbes such as E. coli and Salmonella, which are members of Enterobacteria zeae, into water bodies. Enterobacteria zeae are gram-negative rod-shaped bacteria, which are also facultative anaerobes. They are known to cause infections such as urinary tract infections, gastroenteritis, bloodstream infections, various nosocomial infections such as hospital-associated pneumonia, as well as intra-abdominal infections. Most infections of Enterobacteria zeae microbes are treated with quinolones. And quinolones are synthetic antimicrobials which have a lead compound of nalidixic acid. However, they were also modified by the addition of a fluorine substituent, which led to them having a very wide range of antimicrobial activity against both gram-negative and gram-positive bacteria. Their method of mechanism includes targeting the DNA gyrase and topoisomerase 4, which are very important enzymes in DNA synthesis in the replication processes of bacteria. However, over the years, there has been a wide emergence of bacteria in quinolone resistance. Quinolone resistance in Enterobacteria zeae is mediated by chromosomal mutations, which include alterations to these target enzymes, as well as drug accumulation. There has also been studies that shows an emergence of plasmid-mediated quinolone-resistant genes. All these chromosomal mutations, as well as plasmid-mediated quinolone-resistant genes, can be passed down from one bacteria to the other through horizontal gene transfers. Therefore, this study's aim was to isolate Enterobacteria zeae and to screen for quinolone-resistant profiles amongst them. The study was conducted in 11 villages of Mafikeng, which are listed below, and the two water samples were collected per village. Upon arrival in the lab, a serial dilution of the water samples was conducted, and the water samples were filtered, whereby the filter was then placed on selective media, which is Mekonki agar, which is selective for Enterobacteria zeae, and the colonies were picked and taken for further identification tests. Gram staining was performed, and biochemical tests were performed, as well as molecular test, PCR, was confirmed for the identification of the isolates. The Kirby-Brewer disc diffusion method was used, and a panel of 5 antibiotics, quinolone antibiotics, were used mainly, namely ciprofloxacin, nalidixic acid, levofloxacin, norfloxacin, and ofloxacin. From the study, 35 presumptive Enterobacteria zeae were isolated, and with further biochemical tests, the isolates were narrowed to 13 presumptive E. coli, 12 presumptive Klebsiella, and 10 presumptive Shigella isolates. For molecular confirmation, DNA was extracted, and PCR was performed for the confirmation of the E. coli UIDA housekeeping gene for the presumptive isolates. And 7 out of the 13 presumptive E. coli isolates were confirmed and 6 out of the 12 presumptive isolates were confirmed molecularly for Shigella. However, no PCR was conducted for Klebsiella. However, no amplification was found. With the confirmed E. coli and Shigella isolates, with the confirmed E. coli and Shigella isolates, antimicrobial susceptibility was performed. And it is important to note that only 4 out of the 6 Shigella and 5 out of the 7 E. coli isolates were tested for antimicrobial susceptibility tests. And this is due to contamination that occurred in the lab. The results for E. coli isolates for antimicrobial susceptibility are as follows. E. coli showed a very high susceptibility rate towards the quinolone antimicrobials. However, it is very important to note that one of the E. coli isolates conferred resistance of up to 4 out of 5 antimicrobials that were used. This was a different observation for Shigella isolates, as 75% of the Shigella isolates conferred resistance against nalidixic acid, and there was a 50% resistance towards the quinolones for the rest of the quinolones antimicrobials that were used. In conclusion, the detection of E. coli and Shigella in the groundwater that was isolated shows that the continuous consumption of water in these groundwater in these communities can lead to life-threatening diseases. And also a number of these isolated strains were resistant to quinolone antimicrobials, which is of great medical concern because quinolones form part of the widely prescribed antibiotics for a lot of infections. Resistance towards quinolone antimicrobial is of great concern. Therefore, it is important that there should be a frequent assessment of the occurrence of quinolone-resistant profiles and bacteria in the environment, as well as the establishment of what leads to the development of these resistant strains and the extent that their presence leads to the clinical failure of quinolone treatment. Thank you.
Thank you, Zipiso. So you said that you did this in your honors year. Did you continue your studies?
Yes, I did. And I'm currently doing my master's.
Okay, well done. We'll keep the questions over to the Q&A session. Thank you very much.
Thank you.
Okay, our next speaker is Tuto Magome, who's going to be focusing on microplastic pollution and associated microbial community potentially harboring beta-lactamase genes in a South African river system. Tuto is also from the Noordwes-Universiteit. Thank you very much.
Good day, everyone. Am I audible?
Yes, you are. Thank you.
Okay. Greetings, everyone. My topic for today will be focusing on microplastic pollution, harboring beta-lactamase genes in the South African river system. Now, microplastics, as we know, our river systems have been widely reported to be polluted, and plastics as a new kid on the block is widely used across industries because of its long-term durability and also its low-cost efficiency. However, despite this beneficial use of it, it has become quite a concerning pollutant, and more especially in marine systems. Now, it is estimated that in South Africa, about 15,000 tons, between 15,000 and 40,000 tons of microplastics is carried to the oceans per year from inland sources. Now, what are microplastics? Microplastics are particles that are reported to be with a diameter less than 5 millimeters and not less than 1 micrometer. So it's between 1 micrometer and 5 millimeters. And they can be classified into 2 groups. We have primary microplastics. These are substrates that are manufactured to be microscopic, and they are usually termed microspheres or microbeads, and are mostly produced in the industrial in industrial and household products such as your face scrubs, your shower gels, and your hand cleaners. And secondary microplastics substrates that originate from large plastic material. Whenever we throw them away, they fragment into smaller pieces until they fall within the classification or definition of microplastics. One thing we are aware of is our river systems are reservoirs for bacterial pathogens and also antibiotic resistant genes, as also our wastewater treatment plants are not equipped .
Sorry, I lost the audio.
With that, the growing concern from a microbiological point of view is that microplastics can be colonized by various microbial cells and can maintain stability through biofilm formation in the plastisphere. And also studies have reported that within the microbial consortia, there is an exchange of genetic material between both pathogenic organisms and non-pathogenic organisms within the plastisphere and also within the surrounding water. Now, my objective this actually was the first study to potentially the first study investigating this inland freshwater systems in South Africa, as far as I'm aware. And also the second objective was to characterize the microbial consortia on microplastics from both the microcosm experiment and also in the river system itself. And lastly was to detect and quantify the antibiotic resistant genes from these microplastic samples and the surrounding water. For the microcosm experiment, for substrates were selected, that is 3 types of plastics, polystyrene, polypropylene, and low-density polyethylene, and also wood fragments were used as a non-plastic control. And this substrates were incubated in wastewater treatment effluent and river water. And they were incubated, this plastic were incubated in wastewater for 14 days with 24-hour intervals of isolating one piece to observe the process of colonization. When that DNA was extracted from the substrate,
Hi, Tudu, we have lost your audio.
Oh, sorry.
Yes, you go in and out. I will probably just keep my head in one position.
Oh, okay.
Okay.
Was it here where you lost me? Or just next slide?
Yeah, we can hear you now.
Okay. Yes. So these were low-density polyethylene substrates from both river water microcosm and also wastewater effluent. So when we do a comparison, what we saw was that in freshwater where there's low nutrients, the process of colonization was fairly slow, and only after day 14 we saw a clustering of cells, and most of this was within the areas where the plastic was shearing or tearing and not smooth. And also we observed the same thing with regards to surface properties in wastewater treatment effluent. However, in wastewater treatment effluent microplastic samples, we saw that within day 2 already there is rapid formation colonization of this plastic substrate, and by between day 4 and day 7 already there was already biofilms forming in the clustering of cells, and that's so because that is because to the high nutrients within the wastewater treatment effluent. Now, following that was the characterization of microbial consortia on the substrate. So moving into the environment, first of all, I'd just like to explain that was that 120 liters of water at each site was filtered through 3 mesh steel sieves and to get the plastics from the water. And also these plastics were analyzed to look at the microbial life on the surface using scanning electron microscopy. Following that was DNA extraction or in order to move down further for microbial consortia determination. And lastly, the last objective was the prevalence of MC-beta-lactamase, whereby we were looking at the quantity on both microplastics and the water isolated from those specific sites. And this were the sampling sites. For the microcosm site, the water was collected at site O2 and nwu-4, which is the wastewater. And then for environmental samples, went back again and isolated microplastics directly from the reverse flow filtering process from site M1 at the top and all the way down to process room area. And for the microcosm samples, these were the plastic types we found, we used, and this were bought from a manufacturer. And then within the environment, following the process of isolation, was that we found 4 groups or different types of microplastics, and we grouped them to fibers, films, foams, and fragments, and a total of 206 microplastics were isolated from across all those 5 sites. And following that, what we saw was that the fibers were the most dominant type of microplastics that were isolated from the river. This was followed by fragments. Fragments being used with all those various hard surface types, and then this was followed by foams, which is your polystyrene type of plastic, and then followed by films and microbeads. Following and because in the environment, it's hard to determine when the plastic was introduced, looking at the process of colonization as we did with the microcosm. What I did here was to do a comparison between the life day of the microcosm colonization experiment and looking at also the time from the environment. We looked at the various times where the plastics were isolated from which would you think that it would be the longest time they had been in the water. And what we saw was that although biofilm formation formed within after 14 days, in the environment that the microplastics were heavily colonized by various types of microbial cells. Even we saw a presence of diatoms within this plastisphere. Following that was now the microbial diversity and distribution on those plastics. What we saw was that from the microcosms, the microbial community in the microplastics was less dominant and less even than in wood fragments or in water samples, which means that the water samples had more richer bacterial diversity. And also we saw something similar to what our environmental samples showed us as well, that the water samples still were the most dominant and most diverse than the microplastic samples. Following that was to do a biomarker analysis whereby we look at the microbial diversity distribution. And what we saw was that from the microcosm part, there were some bacterial families that were mostly dominant on microplastics than in the water and also than in wood. And this is basically, we deduce that it's basically due to the bioformation process at the occurs. And also in the environment, we saw that there were some organisms that were more dominant on microplastics and some were more dominant on our water samples. Also quite interesting was that we found some samples where some bacterial families would be solely found on microplastics, although we had incubated what clean sterile microplastics within this water. So that was also quite interesting. With that being said, was to look at the prevalence of antibiotic-resistant genes in the environment and to explore this relationship. We also quantified the bacterial community so that we are able to compare the antibiotic-resistant genes on the microbial community, because it would be difficult to reduce that antibiotic resistance on the plastics alone, because they say the notion is that the microbial community are the ones that carry these resistant genes. And what we saw was that from the microcosm experiment, the microplastic, our microplastic fragments had higher gene copy numbers than wood samples and the water samples. And then in the environment, what we saw was that the microplastics in the certainly from the particular water without wastewater treatment influence was that the gene copies found on microplastics were greater than in the surrounding water community, whereas in the wastewater treatment effluent sample alone, it was quite the opposite. It was the highest. And the next step was to look at the prevalence of MC beta-lactam genes, since they are one of the oldest classes of antibiotic beta-lactams used. And what we saw was that from the microcosm experiment, 3 out of 6 of these MC beta-lactam genes were found, and with that, FOX was the highest on microplastics and in the wastewater and river water, and also this was followed by MOX. And for the DHA, we only found it in the wood fragment in wastewater treatment effluent, and also in the wood fragment in wastewater treatment effluent. Now, when we looked at the environment, the site that was more pristine and isolated from the higher populated area was the initial site 1. From there, we also saw that we found only 2 out of the 6 MC beta-lactam genes. Following that was that there were some highlighted in yellow, but those genes where we found high gene copies in the microplastic samples as opposed to the surrounding water samples where those microplastics were isolated. This was quite eye-catching, because then it suggests the microplastics as vectors and more gene exchanges happening within the bacterial community, within the plastisphere. Also, as highlighted, wastewater treatment effluents contain the highest gene copies. Also, just to highlight here, where we see that from all where the most genes were found, this was where the place is high, the areas are highly populated. And as you can see, as indicated in the red circles, this was from site 2 to all the way to site T5. And this corresponds then with what Dr. Hulof found in the study, where there's high population numbers, there's bound to be high MC beta-lactam resistant genes, and where there is lower high populated areas, and there's also low MC gene copies within the environment. With that, I would conclude that this study served as a pilot study investigating the relationship between microplastics and microcosms and their potential role in antibiotic-resistant genes dispersal. The abundant colonization and selective enrichment of potentially pathogenic microbial groups, and also related to the high antibiotic-resistant genes persisting as gene copy numbers, suggest microplastics as a possible vector for gene transfer and also for antibiotic resistance dispersal. With that, I'd like to unleash the community. Thank you.
Okay, Tutu, thank you very much. You've got a love microscopy that you can see a biofilm on a fiber and a microbead. We will also then keep your questions for the Q&A session. Thank you very much, and all the best with your research. Okay, and that brings us to our final speaker for this session, which is Tabelo Stenger, also going to be focusing on microplastics in the ocean and the potential as a carrier of resistant bacteria and resistance genes from Northwest University.
Afternoon, everyone. I'm Carlos Steiner, a student from the Northwest University under the supervision of Carlos Peseiro and Dr. Odguna Vitmak. My presentation today is based on a review paper that we published recently in Environmental Pollution, which is titled Microplastic Pollution in the Ocean, Potential Carrier of Resistant Bacteria and Resistance Genes. Now, with this paper, we aimed to assess microplastics in the environment and highlight the role microplastics play in the development and propagation of pathogens and antibiotic resistance in the ocean. I believe now we all aware that plastics are synonymous with our daily lives, and it is estimated that between 9 to 14 million tons of plastics are released into the ocean per year. And it is projected to increase, this amount is projected to triple in the coming few years if nothing is done to stem the tide of plastic waste entering our oceans. Now, once these plastics are released into the environment, they slowly degrade to what is known as microplastics, which are plastic particles that are less than 5 millimeters. And according to literature, microplastics are widespread in the ocean environment, with higher densities reported in populated areas and also in accumulation zones, such as those in the subtropical gyres in the Atlantic and Pacific Ocean. Now, microplastics are also found in relatively pristine areas, such as the Arctic and Antarctica. And this also further indicates that no ocean environment is spared from this pollutant. Now, microplastics are not only problematic because of their size, but also because of their ability to absorb organic and inorganic pollutants. And microplastics also serve as an ideal niche for bacterial colonization. And most importantly, they carry pathogens and resistance genes. Now, microplastics in the ocean environment are reported to be colonized by microbial communities consisting of cyanobacteria, Proteobacteroidetes, and beta bacteroidetes and Proteobacteria. And these microplastics can also be found selectively enriched with members of Rhoseobacter, Vibrio, and Pseudomonas. And they are commonly described to be different to communities on naturally occurring substrates or in free-living communities in the ocean. Furthermore, these microplastics in oceans carry pathogens such as Vibrio, E. coli, and Aromonas. And these pathogens have been reported to carry virulence genes, virulence factors such as those for sugar toxins, and also they carry genes which confer resistance to chloramphenicol, fluoroquinones, and also beta-lactams, amongst others. Moreover, microplastic biofilms are shown to selectively enrich various resistance genes, and also they exhibit multi-drug resistant profiles. And in agreement, a number of studies have also reported that microplastics absorb antibiotics on their surfaces, such as sulfonamides and also tetracyclines. And this is tragic because the World Health Organization considers antimicrobial resistance as one of the top 10 global public health threats currently facing humanity. Now, the question still remains, why should I care? Well, we live in an interconnected world, and our actions cascade throughout every ecosystem. Therefore, the plastics that we release into the environment eventually comes back to us full circle. And these plastics do not just negatively affect the environment, they also affect the animals in them. And Also, these plastics affect us humans, and this can be through direct exposure in bathing areas, such as contaminated beaches, or indirectly through consuming seafood which is contaminated with microplastics. As such, we can't hide from our actions, and we need to be conscious of the choices, conscious of the consequences of our actions, which also speaks to our misuse of antibiotics and also our negligence using and discarding plastics. In closing, microplastics are indeed widespread in the environment and also are exponentially growing in the oceanic environment. And with that comes a number of concerns for environmental, animal, and health and human health. Since these microplastics carry pathogens, antibiotic-resistant genes, and also antibiotic-resistant bacteria. This further contributes to the challenges faced with the fleeting effectiveness of antibiotics and the increase in cases of multi-drug resistant pathogens. Now, seeing how plastics affect us physically, this is not limited only to that. This plastic pollution also affects the monetary value which we can gain from the marine environment. And this can be through the blue economy in terms of the profits that are lost in the agriculture, aquaculture sector and the tourism sector because of plastic pollution. Therefore, it is essential to realize the Sustainable Development Goal 14, amongst others, as conserving the marine environment is critical and essential for the future survival of humanity. Thank you.
Okay, Kabelo, thank you very much. If I can just ask if you can keep your camera on, and if I can just ask you just to mute yourself, and if all the speakers for this afternoon can please put on their cameras and just keep the mute button on for me, please, so we can move into the question and answer session. Kabelo, well done on the publication. It's always nice when you get a publication, isn't it? Is that your first one?
Yes, that's my first one.
Well done. You always remember the first one.
Thank you.
And environmental pollution. Nice impact factor. Okay, so if the speakers for this afternoon can please put on their cameras for me, we will move into the question and answer session. And I will just keep my eye on the little hand. And I did not see any questions in the chat, but that's okay because I have some questions. But I will keep my eye on the little hand thingy if it goes up. Yeah. So I'm going to start with Dr. Joe Barnes, if I may. Joe, thank you so much once again for your presentation. You're truly one of the reasons I got into water research so many years ago. I'd like to ask, what advice would you, is Joe online? I don't see her.
Here I am.
Oh, hi. Sorry, Joe, I didn't see you. I'd like to ask, what advice would you give to young scientists who want to, find their passion for science? They don't, so many times students will ask us, I don't know what I need to study. I don't know if I must study my MSc, register for my PhD. What is the best advice that you have ever received and that you think is crucial for students who are embarking on their science career?
What I would advise is to find something that you mind about, something that hurts you or hinders you. Find a problem. I find a number of young people come to me and say, I have the following data. What can I make of it? That's usually a very bad place to start because it means that you've now already got the answer. You just have to find the question to it. That is a real high risk study because you're going to end up finding that you can't do this. You can't say that from the data is existing, nothing that you can do. Find a problem and ask yourself, what what information do I need in order to make to get to an answer or to make some recommendations? What do I need to find out? What evidence do I need if I want to make a court case in science for this? And once your passion for investigating is hooked, it won't let you go. So find something that you mind about, find something that appeals to you, that's on some personal level is important to you, and you're okay.
Yeah, you're right. You've got to care about what you're doing. Otherwise, yeah, it's just science. Okay, thank you very much, Joe. If I can move on to Ruan Puri, I'm just keeping my eye on that. On the chat, we don't have any questions yet. Ruan, if I can just ask you quickly a question. When you say sediments, Ruan, I think biofilms, you know, community engagement, interaction in a bioform. What role do you think bioforms are actually playing in your AMR in the sediment?
Well, I think they play actually almost a big role if you're looking at antibiotic resistance in the bioforms and sediment, because like I said in the presentation, the sediment almost works like a preservative for antibiotic residue, keeping it stable for longer periods, keeping it present there for interaction. So I think it's a very important aspect to focus on, to make sure that, because I feel like that's a hotspot where all the trouble will come from. And we saw it with the anaerobes as well, because sediment, if you now look at underneath the water, it's a very anaerobic, nutrient-rich environment. So we weren't surprised to find the Clostridium there, and we weren't surprised to find that they're antibiotic resistant, but it's still worrisome to realize this.
Were you surprised that the profile of your environmental strains clustered so closely with clinical strains and not the environmental reference that you were using?
No, not really, because I think like eventually all clinical strains kind of originated from environmental strains, because all bacteria are always present in the environment. So I think the niche of the link between environmental and clinical, it's almost integrating now, especially if you look at how the waste of hospitals, for instance, are treated. It's everything is going into the river systems. Our wastewater treatment plants aren't equipped to remove any of the antibiotics that's present in these effluents. So I think that it was inevitable for these two to meet eventually.
Okay, thank you very much, Ruan. Good luck with your studies.
Thank you very much.
Pleasure. Sepiso, I'm going to go through all of you. We understand this, right? So you looked at groundwater. When you say groundwater, are we talking about groundwater in its true sense, or are we talking about an unpiped water supply in the communities?
The groundwater that I was talking about, in Mafikeng, most community members use groundwater like in Jojo tanks. So that was the groundwater that I was focusing on.
So is that rainwater, or do they pipe it up from the...
They pipe it up mostly.
So it's coming from an aquifer?
Yes.
And what are the uses of that water in the communities you saw?
They use
the
them
results
for
are concerning. So I just want to know, what do the people use the water for?
They use it for their day-to-day life, like washing, cooking, cleaning, everything. In most communities, that's what they use it for.
Are you planning on going back and conducting community engagement workshops just to inform the community of what you found?
That's actually one thing that I've been discussing with my supervisor. I believe in science communication, and I feel that it's one thing that scientists should really go back to give feedback to the community, because in most cases we report our work in journals, and chances of community members going to journals are very slim. So we should take it upon ourselves to go to communities and engage. And that's something that I wanted to do this year and last year. However, due to restrictions of COVID, we couldn't do that this year, but I really plan on doing it, and on future work as well.
Brilliant answer. We should take our work back to the communities that we actually sample in, right? It affects them more than anyone else. Thank you so much.
Thank you.
Okay, Tuto, we struggled a little bit with your audio, a little bit, but I did tell you to keep your head in one position, and I think you probably listened. Can I ask you a question? You showed us your, is he not in?
I'm here.
Oh, hi. You showed us your family NGS analysis. Can I ask you a question? Because then you went into your antibiotic resistance genes. What about your genera? Did you find, what are your primary genera that you found, and what was the correlation to the antibiotic resistance genes that you found?
Okay, because I did not directly look at the specific groups because it only came up to look at, but what I found was the Enterobacteriaceae, Legionella, and some Pedobacter genera and species were mostly also dominant on the microplastics and also mostly in the wastewater treatment effluent as well. So I made that correlation based off on the study that I had showed of Dr. Kotia, but because I did not directly analyze that directly, I just made that connection from there.
No, it's a brilliant one, because if I look at your SEM, it looks like you had some pseudo on there as well.
Mm-hmm. Yes, definitely.
Is that part of your future research?
Well, I've completed my master's with research, but I'm currently busy writing a paper on it and trying to also extract more data on it. But I'll be focusing something different for my PhD.
Okay, but good luck. Good luck. Well done. Okay, last one. And then I'm going to go to a question from Prof. Carlos for Joe, but really quickly, my question to Kabelo. Was there a review article that you wrote, Kabelo?
Yes.
Was it a review? Did you touch on the role of quorum sensing with regards to the antimicrobial resistance transmission?
No, we didn't consider that in the review.
But if you're going to be looking at microcosms and biofilms, do you think they'll play a role?
Yes, I believe so. And I think I'll look at that within my current study that we are conducting. So as soon as we collect data, we'll see what connections we can make from that.
Okay, perfect. So Ruan, Tepiso, Tuto, Kabelo, very well done. All the best for your future studies. I hope to see you at a conference somewhere. And now I'm quickly going to ask Dr. Joe Barnes a question from Prof. Carlos Poseidonot. He says, overqualified when you have a PhD in the water sector. What are your thoughts on this archaic notion that many of our students face? I don't agree, but okay. What are your thoughts on the notion that you are overqualified if you have a PhD in the water sector, Joe?
We came from an era numerous decades ago that people did a PhD only when they are on the second half of their careers. They only got the opportunity and sometimes the funds only when they are 40 and 50 years old or older. So that meant that the whole system was geared towards senior people getting the senior degrees. Now we are faced with a very quick change over the last 20 years, 15 years even, that people prefer and manage to finish their entire university education system, the whole lot, up to sometimes, if you're lucky, PhD, before they set off for work. And we are ending up with a work structure system that have got a lot of entry-level posts or more entry-level posts and few senior posts. So we are ending up in a scenario where people have a lot of qualifications getting more and more into a smaller and smaller field of specialization and subspecialization and no work experience. So some things they have to learn that entry-level people have to learn in the world of work, while they have very high specialization in their field and in their topic. So we need to find some kind of a balance there. And I would it's not up to the universities, it's not their prime job, but I would like to appeal, since we are now in this conundrum, to the universities to see if they can't let their people get some work experience as well, even if it's temporary placements in places, so that their CV doesn't only contain swat that, and then swat that, and then qualified with that, and then try and go into the world of work. I faced that with this degree in mathematics, that I was overqualified for being a typist or a telephonist. That was all that was open to me. So I know the feeling very well. And the world of work is only slowly changing. So as an interim step, the best that I can suggest is to see if it isn't possible for those people, while they're doing their masters and their PhDs, to have 2 months here and maybe 3 months there of work experience, so that they can, while they are writing up their results, maybe it's not, there are only 24 hours in a day, I realize that, but it will help them as a step towards being more employable. In the end, it is the world of work that will have to change. But it's no use crying for the moon, we are not there yet. So that is the best that I can suggest, even if they do work that they can do over the internet or crit papers or something, but some experience of some kind. And then there are always, of course, short courses that you can attend in managing staff, in budgeting and things like that, which if you come from a completely focused university study, you have virtually no experience of. So those are steps, small, I admit, that I can suggest to bridge that awful gap between having luxurious qualifications and some gaps that will make it difficult to get a job.
Yeah, I agree with you.
I hope that helps.
No, perfect. Work integrated learning is crucial. I agree with you. So thank you very much again, Joe, for presenting our keynote this afternoon and sharing your wisdom with all of us. I'd also like to thank all the presenters for this session. Good luck with your research. I'm now going to hand over to my colleague, Dr. Eunice Ubamba-Yasso from the WRC for a few closing remarks. Thank you very much, everyone. Have a good afternoon. Eunice, over to you.
Afternoon, everyone. Yeah, we've come to the end of day one of our symposium. I do hope that everyone has been able to stick with us right from the beginning of the program. So my job here is quite simple, just to thank everyone. First of all, our participants, those who sent in abstracts on time for presenting in the two different sessions we had on environmental health and human health. It was really interesting. It was also nice to get presentations from all across South Africa and the different provinces to really see the kind of work that's being done by our students and our researchers. Then obviously to our keynotes, to Prof. Carlos for setting a scene for us, giving us some really good background, good citations, literature, and also really letting us know where we are headed to and giving us the South African and African perspective on the issue as well, making sure that the research that we do going forward is meaningful. Also to Dr. Ryan for his presentation on wastewater studies he's doing in relation to COVID-19. That's a very current set of data that he presented to us, and it's something that worldwide is being developed. So definitely in terms of our research in South Africa, we are also quite on the cutting edge with some of the research topics we are looking at. And then also to Prof. Jo Barnes for sharing with us her experience in the field. I think for all of us, it's inspirational to hear where she started from and where she's rounding up her career. And also for just giving us tips on how to stay not only focused in our sort of skill set, but also in our moral obligations to the science that we do. So I think with that, I would just like to say that we hope everyone joins us again tomorrow. We've got some good presentations lined up as well in the different sections, environmental health, human health, as well as some topics around global recovery. We've also got some very nice keynote speakers who will also be giving us different research topics to think of and different perspectives. And I think that's always important and encouraging for all of us. And then also from the WRC, as the WRC, we are excited to see a lot of our project leaders, a lot of researchers that we fund. And it's also important that our project leaders and researchers know what others are doing, so that when it comes to proposal season, we do see a lot more collaboration. I think that's something that Dr. Nonhlanhla, Dr. Sam, and I are really keen to see. We want to see a lot of collaboration, a lot of bridging of the work that happens in different universities, so that when proposals are submitted to the WRC, it's really reflective of the different research niches that are around the country. So with that, I will not talk further. I just want to wish everyone a good afternoon, and looking forward to seeing you all back tomorrow.